Good morning, everyone. Ready for round two? I'm excited to announce today's lineup. We will be continuing our stimulating presentations and discussions where we left off from yesterday. And for those of you who may not have been introduced to me yesterday, my name is Max Fernandez. I'm a Vice President at Intellective Bio. I have the pleasure of introducing our first speaker for today, Daniel Diaz, the Growth Director of Zymval Biomodeling. He will be presenting an efficient innovation strategy for an effective biocatalysis development. Daniel, the floor is yours.
Good morning, everybody. I hope that everybody is awake already. So my name is Daniel. I am currently working in Zymval, and we are specialized in developing enzymes for many industries, and not just pharma, but more than pharma. And reason why I'm bringing that topic today is that I keep listening that biocatalysis is a very promising process everywhere. But you know what? Reality is basically harder than what we hear, because just a few reactions are made by enzyme. And what's the problem? Where is the gap? Where is the bottleneck if there is a very promising process, but just a few industries are using today? Basically, one of the things that I realized in these 20 years of being in biotech, and again, not just in pharma, is that I have been successfully implemented biocatalysis in cosmetic industries. So we were able to replace more than 46 chemical reactions by enzymatic reactions. And these are the type of results that we get. As you can see, basically, there is a huge improvement between chemical and enzymatic process. Again, this is not a miracle that not can be done in every single reaction and everywhere, but this is exactly the type of benefit of using enzymatic reaction instead of chemical reaction. By the way, in that specific case, esters that were being used in cosmetic companies. Because of the high selectivity of the enzyme, as you can see, there were not being produced any byproduct, so very pure molecule, and that aimed to reduce the amount of energy and cost related to downstream process. Again, long chain and short chain esters has been replaced by enzymatic synthesis. Going a little bit more in detail, that's exactly the benefit that we can see of using enzyme versus classical chemical process. And in that specific case, I'm using an emollient. By the way, myristil, myristate, that's exactly the molecule that we were analyzing. And as you can see, there is a huge benefit of using A versus B, or in other words, enzymes versus chemical process. Not just in downstream, but also in the reaction itself, because we were using much less energy and in much more efficiency. Instead of 18 hours, 6 hours of reaction, to make the same reaction with more pure product. And again, this is not just a single molecule. As you can see, here there is a bunch of molecules in which we were basically successful on making the change between enzymatic process and chemical process. And why? Again, because the productivity of the enzymatic process was much higher. The purity, which is measured by EA, which is acid index, was much less, and that also provide a saving in energy of more than 60%, water consumption as well, waste generation much less, and of course a dilution of the fixed cost because of the enzymatic process. And that's why, and basically this was just because of the selectivity of the enzyme that we were choosing for that specific case. And by the way, as I mentioned, because of the purity, as you can imagine, the product that we were doing that in that specific case, another ester, the purity was very high. And basically that also aimed to the company to not just sell in the current business that they were, example, agro, because they use the same type of molecules, but also in different applications because of the purity that provides a different smell, a different color, and by the way, a different quality for the product. That's another example of what are the benefits of using enzymes versus chemical reactions related to several quality and sustainability indexes. I'm not going to pass to the detail, otherwise we are not going to have time to do that. Again, this is not just in those specific cases that I mentioned to you. We also are helping a company to put SPF booster made by melanin produced by biocatalysis, in which we have seen a very good results in terms of performance and sustainability and competitive price, everything together. This is an example of what we have seen in terms of application effectiveness. In that specific case, melanin, which is very natural for human skin, were providing more than 3 times better performance than existing synthetic SPF booster. And not just that, and that's basically because of the efficiency of the process between synthetic and biochemical process. As you can see, there is instead of a bunch of byproducts produced by synthetic pathway, we were producing just melanin. And that's because, again, of the selectivity of the enzyme versus the chemical pathway that has a much better performance on yield and purity. And again, it's not just about sustainability, it's not just about quality, it's also about cost. In that specific case, there is melanin in the market, which the price is unaffordable for a bunch of applications such as cosmetic. In that specific case, we were talking about 500 euros per gram of melanin. That's the current price in the market, and we were able to drop 1,000 times. Again, this is not a miracle. That's not what we are going to do everywhere, but that's a very concrete case. Also, sustainability index and high quality melanin. So that's the point. I think the point is not about the biosynthesis or the biocatalysis. The point is all about development. That's where the problem starts when we are talking about biocatalysis. And why? Because it's a very dangerous game, because it's necessary a high investment and doesn't necessarily give you or give to companies the prediction of good results. Because the way that we are doing today, it relies a lot on bets. We just go to the lab and we invest a lot, but we not necessarily know what's going to be the result. Even our company, which is, again, a company very specialized with very sophisticated tools to make enzymes, in the computer, or tailor-made enzymes, we were also, even again, working with bioinformatics, molecular modeling, and AI, we were not so successful on developing enzymes for ourselves and for our clients. And at a certain point, at a certain level, we decide to use our capability on molecular modeling, and not just that, bioinformatics, to start to predict the result or the outcome before really starting. And that was the critical point of unsuccess rate to more success rate in the development. So again, not just using molecular modeling, AI, and etc. to make the model, but also to predict the result. That was exactly what we have done. Going to a little bit more on the details on that. That's exactly how we do it. Basically, we make a virtual enzyme screening and in public and private database. Basically, we use, again, bioinformatics and AI to select the sequences which have a reaction similarity of the reaction that we are looking for. Then we take the representative sample of those sequences, and then we make the molecular modeling to go to a more precise vision of how likely that enzyme will work or will perform in the substrate. And based on the docking quality and the frequency of the docking between an enzyme and a substrate, we are able to rank what is the possibility to run the reaction. And this is the type of result that we get. We get a confidence score if the enzyme will run or will not run properly the reaction between the enzyme and substrate. And that gives not just to us, but to our clients, the possibility to make very educated decision on the investment. Should I invest in something which is very risky because there is not a very good fit or quality of the fit between an enzyme and substrate, or should we decide to go something totally different or invest in another pathway because this is not so promising? And that's the result, not just for us, but for our clients on the success rate on the campaigns of enzyme engineering and enzyme discovery, which is very decent. That provides a very confident score, and not just that, a very disciplined decision-making to invest in biocatalysis. And the magic is that because of the decent tools on bioinformatics, AI, and molecular modeling, we are able to do that in less than 1 week. And again, lower investment than going straight away for any type of campaign. And doesn't matter if like direct evolution with high throughput screening or molecular modeling, whatever is the company, we really don't care. And it's the same, it's like a very high investment. You are, at this point, you are able to go for any campaign with more confidence than what you get at the beginning. Of course, we also design and engineer enzymes, and the way that we do it, the enzyme engineering is a little bit different from what currently is being doing on direct evolution or with high throughput screening. We use molecular modeling to evaluate what we call the hotspots of the enzyme. And we propose the changes on the enzyme to have, in order to provide a very good fit between the enzyme and substrate. And that also, with lower effort in the lab, provides a very good confidence level of what exactly is the enzyme that will perform as expected to our clients and to ourselves. And basically, instead of going with thousands of enzymes and producing a lot of mutants, a company and also ourselves, which we develop our own enzymes, we just go with a few to the lab. And again, of course, we use a lot of things that somebody mentioned yesterday, such as AI, molecular modeling and bioinformatics to get those results in several points of the methodology that we use. Basically, what happens on using that approach is that, again, our success rate is very high, and not just that, it speeds up, it reduces the amount of wet lab work, and also, which is different from going straight away for a direct evolution, is that you have the ability to work as a flight simulator, because you can have a very good chance to know in advance what are you changing and why are you changing, and if that change in the enzyme makes sense or it doesn't make sense. And basically that's suitable for any type of enzyme. It's agnostic. It's all about changing the enzyme, and again, it doesn't matter at a certain level. It doesn't matter what enzyme that we are working at, and it's suitable for any challenge in which enzyme can run the reaction. And I'm going to give you some of the examples of what we have done in the past. In a specific case with ADM, I'm sorry to don't bring pharma examples, because pharma is like too hard to share examples with everybody. But in that case, it was ADM, and we were developing an enzyme with a higher selectivity and activity at the same time for them. And basically, there were two reactions. One of them, we started with 160,000. We went to the lab with 60, and we found 18 hits. Same in the reaction 2, but we explored a little bit broader database. We sent to the lab just 45, and we found 4 hits. Basically, you can see that part of the enzymes that we found or we discover for them have a much higher property or function, which in that case, the conversion that we discover for them was 40 times higher than the ones that they have in their portfolio. Second one, it is, and we call our enzyme engineering enzyme volver. We went from, we were working on the active site and distal mutation. I don't know if you are aware of that thing, but it's all about points very close to the active sites and structural points far away from the active site, which provides which actually it's very important for the structure of the enzymes. And we were able to go to the lab with, we detected 81 points in the enzyme that we should improve. We select 90 mutants, and we went to the lab and we found, we work in 20 enzymes, and there was an improvement of 3 times better performance than the current one. Basically, we made the same thing, getting the same enzymes, the second round of evolution, and basically we found at the end of the day, or we designed at the end of the day an enzyme which was 8 times better than performance than the current one. Third case, in which we work in 4 reactions, and by the way, one of them we didn't succeed, and our client was working not just with us, but with also another company, and basically, which was using direct evolution, and basically, they screened 300 enzymes, and they didn't found any hit. In our case, in same amount of time, 6 months, we tested just 44 enzymes in the lab, and we found 3 hits. and that provides a new process with a new enzyme for our client. And that's it. Basically, coupling bioinformatics in AI in a very educated way with molecular modeling generates a very precise data, and depending on the way that you are using those data, it minimizes the risk and also the chance of success of developing enzyme and biocatalysis for not just for us, but for our clients. That's it. I hope that you enjoy the presentation. Thank you so much. By the way, I'm not so technical. Please don't crush me with the technical stuff.
Hi, I would have a question concerning the manual interactions in this optimization process. So where are they?
The manual interaction, there are basically two manual interactions. One, when we are working on enzyme engineering, enzyme design, and basically it's not totally automated identifying the hotspots, right? It's still manual. Somebody or a specialist needs to take a look and to see what is actually not allowing the best fit between the enzyme and the substrate. And then, of course, lab effort is also manual. For the prediction, that's not the case because we just check it out the fit between the enzyme and the substrate, and the fit is the quality of the fit, which represents how much energy is necessary to get the fit. And not just the quality of the fit, but is the frequency of the fit between the enzyme and substrate. That's exactly what we do. And in that specific case, no manual work. Reason why it's done in just 3 days and not in 30 days, which is the common enzyme engineering campaign.
Thank you. And according to your experience, are certain enzyme types more amenable to optimization, or so like oxidoreductases or transferases, or is it the same for everyone?
We are, I'm going to say, pretty much agnostic, which is not exactly true because there are enzymes which are a little bit trickier, but so far we didn't have found so many differences between enzymes, and not just about enzymes. It's all about parameters as well, like thermostability, pH, activity. It's more or less the same because at the end of the day, the technical people they see vectors. They don't see the enzyme itself. They don't care what actually the company is doing, because they will change the structural points, which are physical-chemical interactions between an enzyme or between an enzyme, and that's it. And then they evaluate the external force and internal force in order to hold the enzyme structure and to provide the ability to have the best fit with the substrate in a certain condition.
Thank you. My last question.
Sure.
Sorry. It would be towards, you're optimizing towards reaction efficiency and conversion, but do you also consider the stability of the enzyme, also with respect to being expressed at some point as well, and then needing to be purified, and then the harsher conditions you apply, and then you can apply during the purification, the easier it is then also for the later?
Yep. So good point. So there are two answers. One, it is we work in the whole stability of the enzyme for the development, and again, stability can be solubility, can be thermostability, can be chemical stability. That's for sure what we do. What we are not so far specialized is in the enzyme production. And that's the part of the stability that we don't touch. so far because we have external partners to produce the enzyme in a more efficient way than we do it because we are more specialized on computational development than biomanufacturing, classical biomanufacturing production, and that's it.
Daniel, I'm here.
Okay.
I enjoyed the presentation. First question that I had relates to the intellectual property. When we develop an enzyme with your help, do you keep the intellectual property or do you give it to the client? How does it work?
Yeah, thank you for the question. It's all about who is paying. And if the client is paying, yes, it's client is owner of the IP. If we are co-developing or developing, we are owner of the IP. What we are actually launching nowadays is a customized enzyme kit in which we initialize the work of discovering or determining the most promising enzymes for the client, in which we still have we still have the ownership of the IP, but if the client wants to take one of that enzyme and develop with us and pay for the IP, it's their IP. If not, it's not that case, they just want to be a enzyme user, then we develop the enzyme, we keep the IP to us, and that's it.
Okay. The other question relates to a specific class of enzyme that we call nucleases. So, these are enzymes that are able to interact with nucleic acids. And this is a very busy IP landscape. There's a lot of nucleases. And so the question is, do you also offer some, let's say, IP support in terms of making sure that you're not developing something that is already existing and protected by others?
Yes. We do it, especially like in pharma space, in which companies are very interested on IP, which is not the case for cosmetic companies, by the way. We do that work. We not just find, but we also develop enzyme that is a proprietary enzyme or has a freedom to operate and eventually a proprietary enzyme for our clients.
Very good. Thank you.
Welcome.
No questions from online?
No online questions.
Okay. Thank you, Daniel,
Oh,
for a very informative
it's my pleasure.
presentation, and thank you to the audience for the questions. From enzymes, we pivot back to cell and gene therapies. It's my pleasure to introduce our next speaker, Nick Preya, Associate Director of Gene Therapy CMC Development at UCB Pharma. He will be sharing with us enhancing therapeutic gene encapsulation while minimizing host cell impurities to maximize the quality of recombinant AAV gene therapy products. Nick, the floor is yours.
Good morning, everyone. I'm obviously very happy to be here. This is a very fancy title. I'll try to make it a little bit more simple for you. I realize that the audience is not necessarily expert in recombinant AAV development, but to put it very simply, what I want to do is to share some quality perspective on the development of these viral vectors. So I'll start with a few words about my company. So I work for UCB. We are a global biopharmaceutical company We have a presence in 36 countries, but main activities in Belgium, UK, and United States. We strive to act as a responsible company in terms of sustainability. We are reaching millions of patients. We've got a number of established brands, and we're lucky enough to have a number of products that were recently approved on the market. So you may be interested that we are, in this context, listed on Euronext. And maybe more importantly, we are also reinvesting a significant proportion of our revenue in research and development. For last year, I believe that was 31%. I'll skip the other introducing corporate slides. And I think what is more important is to explain that at UCB, we've got small molecules, we've got biologics, mainly antibodies, and more recently, we realized that it's not enough, especially if we want to address the medical needs of patients suffering from severe genetic disease. And so for that, we are developing now a new pipeline of gene therapy products. And the vision that is behind it is that we want to move from symptom management, which is the case for most of the drugs that you find on the market, move to disease modification, and ultimately the goal for us is to provide a cure in one shot to the patient. We are very serious about this ambition, and so we are investing massively in the development of new facilities. You've got a recent picture here of a new facility. The sky is blue, things are going well, and we should have manufacturing in 2026 at GMP grade. I belong to a CMC organization, so in my context, my specific context, we want to deliver robust, high quality, commercially attractive, and sustainable processes for our product. So gene therapy products are new to UCB. They are also relatively new to the industry overall. I think the first product was in the US approved in 2017, so it's still relatively recent, and we're still trying to figure out what are the quality requirements for these modalities. I couldn't resist to the pleasure to share this anecdote with you. I learned not so long ago that Heinz, the guy behind the ketchup, was championing the establishment of the so-called Pure Food and Drug Act in 1906. That's the ancestor of FDA. And so the reason behind it, or maybe one of the reasons behind it, is that by the time there was a lot of competitors putting on the market product from very variable quality, and some of the products were using very rancid ingredients, resulting in probably adverse health conditions for consumers. And so we need to define quality standards. The quality standards that is inspiring us at this stage are very much derived from guidelines that were initially written for biologics, antibodies or vaccines. And so we realized that they don't necessarily fit 100% with the specificities of gene therapy product. And so we are very active in trying to redefine what are the standards that apply. We do it internally, but we do it also with other companies. And I here mentioned Bioform, where we are very active, and there are also a number of other companies specialized in development of ATMPs, ATMPs being advanced therapy medicinal product. So the specific problem that I would like to discuss today, because there's a lot of problems, is do we have a problem actually with the toxicity level that we've got for host cell DNA, host cell DNA being a process impurities for us. And I would like first to draw your attention on the bottom of the slide. On the bottom of the slide, you can see the 9 recombinant AAV products that were approved, one being freshly approved last week. And what you can see here is that the dose for this product vary greatly. So for an individual of 70 kilograms, we've got in between 10 to the 11th to 10 to the 16th or close to that. So it means that we've got doses that are spanning between 4 and 5 logs. And in the context of our vectors, that means that for a given product, for a given process, that results in a very high load of host cell DNA because there will be a linear increase in between your host cell DNA and your dose. And so on the top of the slide, what I've put is a summary of the regulation that apply in that specific context. And I'll start with what I would call historical guidances. In these guidances, there was a specific threshold to reach, and that threshold is actually very low. Okay, so for most products, including antibodies, this is relatively easy to achieve. It's not necessarily the case for recombinant AAV. And so later on, there was some reconsideration of this specification, and so people are still wondering where exactly we should put this threshold. What I discuss here is what are the risks actually associated with these host cell DNA. So host cell DNA appears during the manufacturing when you rely on cells, and then from these cells you have the host cell DNA that is carried throughout the drug product. So the first type of risk relates to insertional mutagenesis. So you've got and that's an exogenous nucleic acid that is transferred into the cell of the patient. You're not lucky. It goes in a region that will influence the expression of an oncogene or a tumor repressor, and then you've got an uncontrolled proliferation of your cell. You have also possibly double-stranded DNA in your cytoplasm, in your cytosol, and this is not occurring naturally. This is recognized by the SIGA-STING pathway, and that results in inflammation, which you want to avoid. If you've got the same double-stranded DNA in your nucleus, there's another pathway, p53, that, especially when overly activated, will result in apoptosis. And then finally, you could have some random integration in the coding sequence of a gene, and that could result in the expression of a de novo epitope. And in that case, B cells will recognize this de novo epitope and react against this epitope. The good news is that at this stage, these risks, they remain mainly theoretical. We've got so far a limited amount of data, but when we look in the data, we don't see a direct correlation between a high dose of host cell DNA and an adverse effect. Okay? And there is a lot of product. Half of the product that are marketed, they have a very high dose of host cell DNA. So there is still a question mark, and what we try to do is learning, do more characterization so that we can establish correlation and based on that, set specification. I'm sure you're not super familiar with host cell DNA, with recombinant AAV, and so there's here a graphical representation of what are the different entities that we have to deal with. On the left side of the panel, you've got what we want to develop. This is a capsid, and in blue, the transgene that has a therapeutic effect. In the middle panel, you see the very same desired product, but surrounded by host cell DNA outside of the capsid. This is a good case scenario for us because it's relatively easy to remove through the process. What is more complex for us is the right panel, in which you have a mix of host cell DNA and product, or just host cell DNA within the capsid. So I'll discuss first let's say the best case, or the easiest case for us in terms of removal of process impurities. And so I show you here two examples where we're applying, let's say, classical approaches to remove host cell DNA. We are using a nuclease here that we refer to as endonuclease. In that case, increasing concentration of enzymes lead to very efficient reduction of the level of host cell DNA because the enzyme, the host cell DNA is accessible to the enzymes. Next to that, we've got a classical affinity chromatography approach in which our vectors are binding to ligands, affinity ligands, and the host cell DNA is not binding, and so it's going through the wash. And again, we've got a very efficient level of removal. Keep in mind that maybe we're several logs over the threshold that people may want us to achieve. Here it's the case that is a little bit more complex for us, and so what you see on this graph is a typical anion exchange chromatography that we are using during our polishing step. And so in this specific setting, what you can see is two main peaks. The first peak is what we consider is the empty particles, so these are capsid without our therapeutic gene. And the second peak on the right side is our desired product, where we've got a high proportion of what we call full capsid. So this is the capsid plus the transgene. When we do fractionation of this AEX, and it's a bit cut here, but what we observe is that there is a much higher proportion of host cell DNA in the first peak here. And so our challenge here will be to find the sweet spot in between getting high yield or high purity. And so it will be somehow a threshold in between a trade-off in between the two. Obviously, what we want to achieve is having high yield and high purity. So we went back to the blackboard and we resumed that, through the purification process, we achieve the best we could achieve. And so we need to influence the process at an earlier stage. And so we look into our upstream process, what else could be done. For those not familiar with this type of processes, we're using cells. We're growing cells to production stage, and at the production stage, we're using a transfection to induce the production of recombinant AV, and we're using combination typically of 3 different plasmids. First plasmid contains the gene of interest. The second plasmid is responsible for the expression of the capsid and the replication machinery, and then we've got some helper functions. This slide is a little bit complex, but I'll try to make it a little bit easier for you. Essentially, what it says is that there is a very complex interplay in between the different components that we need to manufacture this product. I'll take just one example to exemplify this complexity, and I'll have a bit of water. So, one element creating a non-specific packaging of nucleic acid here is the relative proportion of some of the rep components. So what we refer as rep is a set of different peptides, and some of these peptides will induce a sequence-specific production, and some of the peptides will induce a sequence non-specific. And so the relative proportion of these entities will definitely influence what is packaged within the capsid. On top of that, there are many other technical avenues for us to, from a genetic point of view, influence the proportion of host cell DNA versus transgene that we are able to package or not. I will not go into the detail, just show you one specific example where through engineering of our plasmid, in this case helper plasmid, we are able to remove, in this case, 30% of the host cell DNA without compromising on the level of productivity that we have. Is it enough? From our perspective, not necessarily. And so we wanted to explore other avenues because it's a lot of energy to generate plasmid constriction. And so what we did is, based on the observation that some of the components involved in the manufacturing of recombinant AAV, they induce a level of toxicity. And this is typically what you see during the production. You've got a drop of viability as early as you induce the, when you have the transfection. And so what happens is that in the nucleus, you've got the packaging of the transgene or cell DNA, and at the very same location, you've got a degradation of the genomic DNA. And so this creates the perfect condition for packaging unintended nucleic acid. So what we did is we've used a caspase inhibitor. And in that case, during the production, you see that results in an increase of the viability of the cells. And concomitantly, we are seeing with the very same inhibitor a drastic reduction in the level of host cell DNA that we see at harvest, but more importantly, post-affinity. The use of pharmacological inhibitors is not always desirable because it will involve a lot of additional step of purification and demonstration that you can clear the reagent that you have used on your process. And so for us, the next step is probably cell line engineering with this inhibitor as a proof of example. So I think as a first approach, we have tried to focus on host cell DNA load, but probably the next step for us is also, is there a link in between the size of the DNA that is packaged, the host cell DNA that is packaged, and the specific sequence. Sometimes it could matter, sometimes it doesn't matter. And what we consider is a higher risk here is the introduction of oncogene in your product. And so you've got two kinds of oncogene. You've got those that are from viral origin and those that are from human origin. Those from viral origin, they are quite well characterized. So for ACT293 cells, you know that you will find E1A. On the other hand, for human origin oncogene, there's a very long list. So the story here is a little bit more complex. So what I did is I had a look at top genes that are involved in oncogenesis in human. And if you focus first on the coding sequence, you observe that most of the gene, the coding sequence of most of the oncogenes, they have a size that is compatible with packaging in the genome. The good news again is that there is a very big difference between the size of the coding sequence and the length of the gene. So that means that the likelihood of packaging those genes is virtually impossible for most of the oncogene. On top of that, you not only need to have the oncogene that is packaged, you also need to have a double-strand break in your DNA so that the oncogene is inserted in your genome. So we're talking here about very unlikely events. So I'm already hitting the last slide of my presentation. And so I wanted to quote one of our local philosophers in Belgium, and I think he was very right saying that he had his fair share of bumpy roads and heavy winds. That's the very same for gene therapy. There was a lot of clinical adverse event that sometimes for several years had negative consequences on the whole field. So I think we hold collectively a responsibility in making sure that we are developing high-quality product because each time there is an adverse event for this innovative product, the whole field is challenged, and that starts with financing of these very expensive therapies. So the problem will remain for a while that we've got very high level of cell DNA for high-dose gene therapies. And what I've tried to explain today is that there are possible mitigations through production, through plasmid engineering, through cell line engineering. We can talk also about supplemental characterization and animal studies. And all this is part of an integrated strategy, a risk-based approach strategy that will help you to set your specification, which will help to define the safety of your product. And with that, I thank you for your attention.
Very nice presentation, Nick. I've never heard anyone refer to Jean-Claude Van Damme as a philosopher. I'll keep that in mind. No questions from online? Camila.
I would like to reconnect to your question from yesterday. Do you think that PAT can help you during your work, and in case, in which steps?
So PAT is, we see PAT as not an option, but a must in the development of our processes. We are included at this stage a number of PATs. There is a number of PATs that help us to support the development of the process. Later on, possibly manufacturing as well. And we're still at a relatively early stage. And so the way it works for us is we go to conferences, we look into the literature, we identify ideas that we collect in a file, and then with the leadership team, we have a look at those ideas and we say, okay, we've got a lot of ideas, what is really creating value for us? And the technology that you have presented yesterday is already in this file. I did it yesterday. So it's something that we continuously review, and then we try to assess in our development laboratory, and then hopefully it will help us to develop higher quality product on which, with processes on which we have a higher level of control.
Let me ask what may be a naive question. So the guidelines were developed with total lifetime exposure in mind. Biologics are usually given over several years, whereas with cell and gene therapies, these are usually 1 or 2 doses, correct? Has there been any discussion regarding total lifetime exposure to host cell DNA and other process impurities in setting these limits?
So I actually went through these different guidelines that I mentioned in the presentation, and they don't address the point of recurrent injection, because first they were written for product with recurrent injections. in the context of whole cell DNA, I'm not sure that this is 100% relevant because we try to develop therapies for which it's one injection. And maybe on that specific context, it matters less that we have, I mean, it will, from an inflammatory point of view at least, we should be able to have higher levels of whole cell DNA because the secondary responses, inflammatory responses, usually is more aggressive. But those responses, they are very often product specific. Here, whole cell DNA, we believe, will not be specifically packaged. And so when we refer to whole cell DNA, this is very generic type of molecule that does not induce, we believe, a specific immune response. It's more the innate immunity that I believe is involved in this case, but it's a very relevant question.
Thank you, Nick. One more question. You focused specifically on host cell DNA. Are there any other process impurities that could present a risk to patients?
We believe so. And so on top of host cell DNA, I think one critical element is characterization of plasmid, because plasmid could lead to the recombination to a natural AAV. And so this is something that we are routinely testing, and we are optimizing also our plasmid construction in a way that we observe much less frequency in what we call reverse packaging, which means that a nucleic sequence of plasmid is packaged within the vector. I could continue with more classical, let's say, process impurity, like all the elements that you put during your manufacturing, your enzymes, your reagents, and so on. But more specifically, I think we have a focus on genetic elements, because those genetic elements we know will remain for a while and represent a risk in terms of integration in the genome, which we really want to avoid.
Our next presentation will be a virtual presentation by Taha Abdulwanis, who is the Senior Biosimilar Production Specialist at BGP. He will be calling live from Egypt, I believe, and the title of his talk is Harnessing AI and Machine Learning for Next Generation Biomanufacturing. Taha, can you hear us?
Yes. Hello.
The floor is yours.
Yes. Let me first share my screen. Oh, wait. You are good now? You all see my screen?
Needs to be in presentation mode.
It's good now? Okay.
Yes.
Hello, everyone. My name is Taha Abdulwanis. I'm from Egypt. Today, we will talk about harnessing AI in biopharmaceutical manufacturing. But before we dive in, let me share a quick fun story between me and my AI chatbot. It's now winter season in Egypt. I told him that I am freezing because it's 90 degrees Celsius. And it responds to me. It's hilarious. Let me share the response to you. Are you hear the response now? Okay.
Hear anything?
He said, huh? There is no sound, I think. There is no sound.
There's no
He
sound.
he said, or it said, act normal, mate. Grab a beer and have a chair beside the canal or something like that. It's a summer season. If you told this to people in Amsterdam, it's a summer season. Then start when the temperature reach to 60 degrees Celsius. So I'm working as a production specialist, senior production specialist in biogeneric pharma. Let me give you a quick overview about my company. BGB is a new state-of-art biopharmaceutical facility specialized in manufacturing of therapeutic recombinant protein and preventive vaccines. We have three main production line, upstream and downstream line, formulation, fill and finish, have two line, biosimilar line, it's called combi line, and vial line, it's stainless steel traditional line equipped with SVB, high-tech SVB, and high-speed filling machine. I will start with the combi line. It's a combi come from combination because the filling machine for this line can fill a three type of container: a pre-filled syringe, cartridge, and vial. And the formulation for this line equipped with single-use technology. This make us adapt with a new product, reduce the cost for cleaning validation, and reducing the time. And there is a packaging line and labs, of course. Our main activity was research and development, production, analysis in our lab, and marketing. As it's shown here, let me zoom. Biogeneric pharma nominated by WHO to receive a platform for mRNA technology to be one of 15 factory in low and middle income country to receive this mRNA technology. Actually, I updated the presentation today morning to add this because this is as an initiative by WHO to make sure that the vaccine goes or reach to local and middle income country. As we said here, in Africa, there is South Africa, Kenya, Egypt, Tunisia, and Senegal. And for Latin America with Argentina and Brazil, and Pakistan, India, and so on. And this on an example for one of initiative by WHO. And on country level, there is South Korea through International Vaccine Institute and its national university has invested in low and middle income countries vaccine professional. By giving them hands-on training, I participate in two programs called GTHB and GXB for biological manufacturing and development. Singapore, as another example, give us through Hillman Lab and development countries vaccine manufacturing network a tech transfer course. I participated in this course. It was fascinating last year. And I must say that Africa vision in 2030 to manufacturing 60% of vaccines that the African continents need locally. This is a huge step for vaccine reliance and self-manufacturing. Yesterday, we shown in Egypt here the launch of Egyptian Vaccine Manufacturer Alliance. This alliance between top local manufacturing for vaccine in Egypt and Egyptian Drug Authority. This alliance aims to provide or reduce the cost or provide a free training for Egyptian vaccine manufacturing. Like a collaboration between regulatory and vaccine manufacturing and research institutes. And I must say that Egypt in 2022 has recognition as maturity level 3 for vaccine or by WHO in 2022. And this initiative is like a small step or like a global direction for empowering the local and middle income country for vaccine manufacturing. To be optimistic, small effort or small change in capability or capacity expertise can profound a huge effect during health crisis, God forbidden. Now, let me zoom out. Our table of contents, our roadmap for our presentation today, we will start for introduction AI, then current challenge in biopharma, key for AI application in biopharma. We will dig deep for key application in real study and example. Strategic impact for when the AI applicable in biopharma, future trends that shape the industry, and question and answer. First, we will talk about introduction to AI in biopharma. What is the AI means? Actually, I prefer a definition that it's a computer system capable to do like a complex task historically that only human can do those tasks. So how AI, we will get back to this definition when we go to talk for the key application. How AI fits in biopharma. AI can be a tool for improving the manufacturing process, make it more precise, make it more faster, decreasing the cost. This through our key applications. This is a quick poll. Please scan the QR code here. Do you use AI in your organization as a professional for your organization or a personal use to help you in your professional career? I will share the live poll, please. I prefer the live poll in online presentation because I'm not here, so I try to interact. Wow.
I think you've got the numbers. 9 yes,
Yep.
3 no. 10,
Yep.
10 yes.
10 yes, 3 no. Let me get back to the presentation. Actually, for 10 yes and 3 no, it's a quite fascinating. We will talk to, now we will go through current challenge in biopharmaceutical manufacturing. First, we have supply chain disruption. Global disruption continue to impact the pharmaceutical supply chain. We, in our company, we encountered delay for single-use bags that for Compiline from 6 months to 12 months. Regulatory compliance, stricter regulation demand more accurate timely reporting because biomanufacturing for some regulation are a mystery. They want to know more, so they ask more a lot of questions, and some of question we don't have the answer for it. R&D cost, cyber security threats, and sustainability. Pressure to reduce environmental impact and adopt green practice, more green practice. Then the key application for the AI in biopharma. I will talk for about 4 main key applications: deviation analysis and predictive analytics, process automation and data integration. we will start for the deviation analysis. This project, actually, I participate in a project like that. Let's consider a scenario where our facility encounter 100 deviation annually as an example. Imagine how much time your team would spend analyzing for each deviation, defining the root cause, initiating CAPA, and after initiating CAPA, they evaluate the CAPA effectiveness regarding the potential for human error for identifying a root cause or relying on unnecessary or non-correct data. And the time of resource required become even greater for considering the human error. So the AI now how can the AI streamline this process? The AI, we will take like a 3 steps for that. First of all, we will centralize the data analysis. AI will review all deviation, categorizing the deviation by the type, frequency, impact. Then using the historical data pattern detection and recurring issue for instance, they will like a root cause prediction from using historical data and the pattern detection. After that, it will provide us a specific corrective and preventive action recommendation based on the analysis from the above steps. And then the AI adapt to a new data, refining its algorithm to stay ahead emerging risks and ensuring recommendation for the remaining relevant and effectiveness. But by the replacing manual process with AI-powered automated analysis, team can uncover deep insight through tool like NLP or natural language process. This tool can analyze the heavy text in C reports or non-conformity report, linking linking unrelated or apparently unrelated deviation to give a clear picture for root cause. The result of this transformation, the time for deviation itself can be decreased to 50%, enhancing operational efficiencies. AI keep updated and can predict you to make a preventive action for incident could be happen in the near future. This is for AI for deviation analysis, and the impact for this, actually I encountered one of these projects. The impact was we save time for like 60%. Now we can focus on the effectiveness of CAPA. We can focus on the real root cause. It's give us a lot of time to focus on what really matter for the deviation. After that, predictive maintenance in the industry. Imagine that like you have a filling machine. This filling machine in Israel area have encountered like some in our machines there is some like a heating frame for heating the tubs. That like to make the glue easier to say the first cover for ready to use tubs. Imagine that the heating frame have a problem for like the temperature itself drop during you work. This is unplanned equipment failure can affect on your process, and there is intervention to solve this or to make the maintenance for this failure or an unplanned equipment failure. This what we called a predictive maintenance in AI. How can AI make this process easy or can overcome this challenge? There is a sensor, IoT sensor, continuously monitors the machine health in real time, tracking parameter like vibration for the sorter, pressure in the valve, temperature. If there any early sign of misalignment, pressure fluctuation, temperature drop for the heating section, the system flags and give it recommendation to check. The predictive analytics is the algorithm analyze all the sensor data, identifying these abnormalities that could lead to failure for this machine. AI, like what happened in our machine, he detects that there is some kind of heater in the frame, the temperature not stable on it. So this lead to smart scheduling for maintenance program. Maintain schedule or dynamic optimized based on this data instead of the following routine or reactive maintenance. Intervention now can be proactive before the equipment failure itself. This can lead. Wait. When we do this as a company, there is a 20% reduction in downtime, and there is a 30% saving in maintenance costs. By smart scheduling and predictive insight, drove a significant reduction in operational cost. Now we will go to the third key application, quality control and real-time process optimization. The problem here, and this is from Amgen, they use this, but let's imagine a scenario that an vial inspection machine inspect from 400 to 600 container per minute, which like equal 24,000 vial per hour. The machine goal itself, it's identify particulates, cracks, cosmetics defects, and injectable solution. But there is always some kind of false reject rate from 10% to 20%. If we look to this percentage, this would result in 4,080 container reject per hour. You need to re-inspect this container manually, and the capacity for an inspection girl in hour is like 600, from 400 to 600 vial per hour. So what the machine inspect in a minute, the inspection girl make it in an hour. So it's like a requirement, a huge time for this. Now, the vial inspection machine integrated with deep learning AI. The AI-powered inspection system use some advanced algorithms that process image like a human precision. This system can distinguish actual contaminants such as a particle from being an elements like air bubble, which are manually or traditional rule-based system might misclassify. By improving the detection accuracy, the false rate dropped to 700 or 70%. We now can ensure the defective product are reliability, identify and remove to minimize the risk. This also gives us a real-time data processing because the machine itself equipped with line scan camera continuously analyzing the image in real time. It's provide instant feedback and makes a real-time adjustment, ensuring constant quality control across the production line. This impact, as Amgen like share, 60% reduction false reject and 70% increase in particle detection accuracy. Let's see. As we see here, 70% increase in particle detection and 60% reduction in false reject rate. Sorry, this is another poll. How ready is your organization to implement AI in biopharma? Hmm. Multipressive. Okay. Thank you all. After that, we will talk about the future trend in biopharmaceutical industry. We will first go to the generative AI. Generative AI create a molecular structure, expediting drug discovery, digital twins, virtual simulation for process testing, and the third one is autonomous personalized medicine. AI tailors treatment based on patients' data. We will talk about this in details. The generative AI, AI in drug discovery, AI can reduce drug development costs by up to 50% and shorten development time by like 30 to 40%. By gathering the real data and analyzing them with advanced AI model, clinical trial enhancement, AI helps improve patient stratification, resulting in 60% faster trial process and 35% reduction in patient recruitment by analyzing the historical data or health historical health for the patient itself. AI now using by cancer treatment enable early detection and personalized treatment, improving early cancer diagnosis by 20%. Multi-omic data integration. This is by combining genomic and proteomic and metabolomic data enhanced disease understanding and leads to like 60% accurate disease detection. Precision medicine AI driven analysis of multi-omic data and expect to improve therapeutic effectiveness by 40 to 50%, and personalizing treatment for better patients and outcomes. Now I am finishing my presentation, but at the end, I would like to say the AI is like a tool. It's a new tool. When we deal with the AI module, we take the validation steps till end. The AI just takes the input data from us and work on it like a small child. And the doctor said to me once, if you give like a child when he see a horse and you say to your child, this is a giraffe. Each time he see a horse, you say this. Your children are alone and said a horse, they will say this is not a horse, this is a giraffe. So AI now is a tool like rely on our given input data. We must validate the input data, because if the data we input shitty data, we will give like a output not our the desirable output. Thank you all. Sorry for like I'm little nervous a little bit. Sorry for my English has some trouble, and thanks for you all. Now I'm finished.
No worries, Taha. We could understand you very clearly. Let's open the floor to questions. No one from online? Well, let me start. Going back to your case study regarding use of AI in visual inspection, can you clarify, is this already implemented for a commercial product?
Till now, it's in development stage, but the result is for the development stage, not for commercial project. They now in the validating the result.
Okay, can you comment on the progression of this application towards adoption for commercial product? And are there any challenges that you and your organization foresee in the acceptance by regulatory authorities?
Actually, for our regulatory authority, it's might like you must like explain this tech first to them, because in for implementing the AI, they have must a lot of questions, like the result that you rely on, the data you have input, how you ensure that the cam itself, the quality of pictures that you provide to the AI module, how can you ensure that he says it's an air bubble, not like a particle. So it's a like it's a like small step for improving the process, but it will take a long time or a lot of time to explain or to implement it quickly, and you to implement correctly. And you must ensure that, because it's a commercial and it goes to the patient, the correct or the good vial are good, and the bad reject are bad. So it's look, it's not like a one-day process. It will take some times to rely on the result of the AI module for this.
Okay, thank you. Let me ask a follow-up question. I'm not an expert by any means in AI, but I assume that there is some training involved with this tool, and maybe that starts with your visual defect library.
Yep.
So can you comment on the steps that your organization is taking in training the tool, and what sort of visual defect library or libraries are you using?
First of all, we will start with like an easy way to achieve like a cosmetic defects in the appearance of the outside layer for the vial line, for the vial itself. Then we will go for like particle. Then we will go for air bubble, some cosmetic defects in the alucab or something like that. Like when you validate the inspection girl, you give it like you make some a type of defected vial, put it in combined with a good vial. Then you see the level of detection for this. This is what we make for the AI module.
One quick clarifying question. Is this application for PFS? It wasn't clear.
Actually, no, it's for vial, but we can apply it for PFS because it's the same process.
Right. Right.
But
I think
actually,
that would,
huh? Okay,
yeah,
okay.
I think PFS would be a natural application.
Hmm.
Barry? Question?
Yep. Hi, Taha. Thanks for the presentations. Very interesting. And you seem to be ahead of the curve in terms of most manufacturers. Do you develop the AI applications in-house, or do you partner with another company?
It's all partnered by another company.
Okay. And
It's
how
all
long did it take to get from, concept to actually, producing good results that you could depend on?
it's take nearly 6 months to a year for providing all the reliable data to rely on.
Thank you.
Any other questions? Okay, Taha, no further questions. We very much appreciate your calling in from Egypt. Have a nice
Thank you.
day, and thanks again. I'm pleased with the opportunity to present to you this morning our commitment, shall we say, to increasing access to biopharmaceutical products. By way of introduction, my name is Max Fernandez. I'm a Vice President at Intellective Biologics, which is a contract development and manufacturing organization based in Suzhou, China. Well, that's interesting. I don't know what happened to the format. It looks completely messed up. But let's continue anyway. So as you could tell from my impassioned comments in yesterday's panel discussion, I am very much a believer in increasing access to biologics. We've had a long history with recombinant protein technology dating back decades. But the truth of the matter is that only 10 to 30% of the global population has access to what we would consider basic monoclonal antibody therapies. What I'd like to do today is to share with you how our organization is operating within a very restrictive framework in China to be able to increase access by driving down COGS through simple measures. We're not talking about implementation of advanced technologies, such as what we heard from Stam and what we heard from continuous purification. There is, I think, a progression towards implementation of these technologies, hopefully in the near future. But what you will see today is something more basic. I'm going to start by talking about the business and regulatory environment that Chinese companies have to operate in domestically. You have to understand this background and what is perhaps a black box for most folks before I give a very brief company overview and eventually case studies. So, as with most products in China, it all has to do with economics and pricing pressures. The healthcare market is a tender market with pricing dictated by the central government. There is what is called a National Registration Drug List, or NRDL, and in order for any therapeutic product to be approved on this NRDL, there is a certain price point that all therapeutic products must adhere to. It doesn't matter whether it's a small molecule or a biologic. There is a price set by the central government. And in many instances, this price is offset by what is called volume-based acquisitions or volume-based pricing. Low price, high coverage is what healthcare is all about in China. You'll see here that there's a valley of death that biomanufacturers have to cross, going from development to commercialization. Because the truth of the matter is that in the last decade, there has been a decrease in the price of biologics by as much as 90%. However, the production cost of local manufacturers for biologics, including CDMOs, has not been able to keep up with this downward pricing pressure. There are two kinds of biomanufacturers in China. There are the innovators with excess capacity who have branded themselves as contract manufacturing organizations. There's a second type of organization, of manufacturing organization, which is a pure CDMO, which is an organization that has built capacity and capability not for their own products, but for the products of their clients. In both cases, all of these manufacturers are under intense pressure to lower COGS while maintaining quality. So there is an opportunity to apply what I consider to be right-scale manufacturing efficiencies to reduce the COGS. Now, as I said, the China regulatory and business landscape is somewhat unusual. And it starts with the commercial market. There is no blockbuster products in China. There are no blockbuster products in China. You contrast that with the international market, meaning in China, what we call overseas imported biologics, where the market is dominated by big pharma and, of course, by the blockbuster products. So in China, because the market is fragmented, there is intense competition, not just for first-in-class products and best-in-class products, but also well-established therapeutics. So that's the number one challenge. In terms of exclusivity, there is some IP protection, but the problem is in China is that once a product seems to be commercially successful, there are 6, 8, 10 other companies that will look to commercialize their own products to compete. Case in point, the PD-1, PD-L1 space in China. There are at least 8 products competing for the total domestic market in China in this particular therapeutic area. Now, there are certain regulatory requirements that are not imposed anywhere else in the world that the NMPA, the China National Regulatory Authority, imposes on domestic manufacturers. And I put here a few examples. First of all, commercial manufacturing of DS and DP, drug substance and drug product, must occur at the same site. Contrast that with overseas, where DS is manufactured in one country and exported to another country for fill finish and final release testing. In China, for process validation, process performance qualification batches, PPQ batches, cannot be sold. Innovators take a big hit if PPQ batches cannot be sold, whereas in the rest of the world, there is no issue with selling PPQ batches. Keep that in mind, because most solutions right now to increase access and decrease COGS rely on economies of scale and increasing manufacturing scales as much as possible. In China, for process validation, the PPQ must be performed in a single bioreactor. There is a concept of like for like, but it is not yet widely accepted. And lastly, during the pre-approval inspection, this is for commercial product, no other products can be produced in the same manufacturing suite. This is not the case anywhere else in the world. So you have these limitations. And the challenge now, how can the biomanufacturers differentiate themselves? What is the sweet spot? There can be no differentiation with quality because all products must meet certain specifications and quality requirements. there's really no differentiation in timeline because there's only so much that one can do to compress the process in terms of manufacturing. You have to have a certain amount of stability data. You are limited by cell culture processing times. And so what's missing here at the bottom is the only differentiation is really in terms of reduction of COGS, which is pricing, and risk mitigation. So the key considerations that a biomanufacturer must pay attention to are facility design, meaning scalability of manufacturing and the flexibility in scheduling. Being able to employ high productivity cell lines that are stable. And thirdly, being able to establish a supply chain ecosystem that is both predictable and is able to control costs. With those limitations in mind, I'd like to spend a few slides on the background of our company. Like I said, we are a pure CDMO based in Suzhou, which is about 100 kilometers northwest of Shanghai. We have 3 facilities. The company was established in 2018. And in 6 short years, we have grown from our initial site in Shangcheng, which was purely for contract development manufacturing for clinical projects. In the middle of the pandemic, we expanded to Suzhou Industrial Park, where we have 8 2,000-liter single-use bioreactors from Cytiva. And finally, our headquarters, which became GMP ready and operational in early 2023 in Changshu. We currently have 29,600 liters, which is a combination of single-use and stainless steel bioreactors online, with a total design capacity of up to 192,000 liters. That doesn't seem like much in today's world where we have giants like Samsung, where every year you hear them employing 150,000 liters, 200,000 liters additional capacity. But I would argue that there is a case to be made for what I call right-sized manufacturing. Many companies claim to be one-stop shops. In our case, what that means is that we focus strictly on antibodies, and we have the capabilities from process development, which includes cell line development, upstream and downstream process development, formulation development, scale up of existing processes, as well as for commercial products, process characterization and process validation. I'm proud to say that 90% of our analytical and characterization studies are done in-house. This includes all methods, not just for protein characterization, but also for stability studies, as well as compatibility testing for clinical products. We have the ability to develop conjugation processes for antibody drug conjugates. We are able to scale up now to kilogram quantities, and we have the associated analytical testing and clinical and commercial manufacturing capabilities in-house. As I said, we focus strictly on recombinant protein therapeutics, which includes mostly antibodies. We have manufactured a few vaccines, especially in support of the pandemic. And we have several ADC projects that are ongoing. As you know, ADCs are a very hot topic right now. We have aseptic fill finish capabilities, not just for liquid filled and lyophilized vials, but also for pre-filled syringes and cartridges. And you will see that my title is Global Regulatory Affairs, but because 90% of our clients are domestic China, most of my time is actually spent outside of regulatory affairs right now. Summary of our project experience. I'm very proud of these metrics because you can imagine being established in 2018, really not much was going on in terms of being able to submit products for regulatory review. So in less than 6 years, we've completed 158 total projects, divided here, like I said, more than half are antibodies. ADCs are another growing area for us, about a third are ADC projects, with the rest divided between complex recombinant glycoproteins and fusion proteins. 381 GMP batches produced, ranging in scale from 200 liters to 6,000 liters. We have not had any overseas regulatory agency inspections, but we have been inspected extensively by the NMPA and the Center for Drug Evaluation, the CDE, at the national level and at the provincial level. As of October, we have had 84 IND clearances and approvals, with no approvals delayed because of a clinical hold for CMC issues. So essentially 100% success rate for the CMC part of the INDs that we've supported. And our clients have submitted to the US FDA, the China NMPA, Australia TGA, MedSafe, as well as a couple of products in the EU. We have currently one commercial product with another commercial product in review, and our client is expecting to get approval for that product this year. So scalability and flexibility starts with the right facility design. It looks like I'm running out of time, so I'm going to speed up a little bit. This is our main manufacturing site in Changshu. We are self-contained. We have a main production building that houses our antibody production facility and a production annex that houses our ADC workshop and our pilot plants for our clinical projects. So we talked yesterday about increasing productivity. And I think it was Martin who made the comment that 1 to 3 grams per liter titer is low. The current status right now is that we can very easily achieve 6 to 8 grams per liter. And what that means is a reduction, corresponding reduction in bioreactor scale from 15,000 liters to 6,000 liters. And our concept here is to have 8 manufacturing lines, 8 purification lines. And there's no one else in the world that I can think of that has 8 purification suites in one facility. Purification is a bottleneck. So I told Art yesterday, I'm very excited with continuous downstream purification, being able to remove this bottleneck. 32 6,000 liter bioreactors, 2 suites online right now. Because purification is a bottleneck, we can assume that there is perhaps 1 batch that can be purified per week, 52 batches. Simplistic calculation here is 6 grams per liter at 65% yield of DS, yields 23 kilograms of DS per batch, or 1,200 kilograms per bioreactor per year. So not bad. The advantages, of course, of scaling down to 6,000 liters is that we reduce our overhead costs. Supply chain. We've heard from several companies regarding how we can improve both upstream and downstream. We are very much focused on our cell culture media to increase productivity. Very excited also to be able to employ new technology for bioreactors, which we believe is another driver for decreasing COGS. We heard from our colleague in New Zealand about a new purification column. So that's another exciting area. And we also partner with both local vendors as well as international vendors to be able to maintain a regulated and predictable supply of consumables. So let's dive right into the case studies. 3 projects, A, B, and C. A couple of them are fusion proteins, PFS, dosage form, and 1 monoclonal antibody, which is liquid IV for injection. The first case is a manufacturer established the process. The client transferred from that initial CDMO to us at 200 liter scale, remaining unchanged, with the only change being in the container closure system, moving from vials to PFS. And what the client wanted was for phase 3 all the way to licensure, changing from the initial CDMO to us, but increasing the scale from 200 to 500 liters. The last phase would be a post-approval change where we would scale up from 500 liters to 6,000 liters. And our approach to this is to optimize the process at 3-liter scale, confirm the process, and lock it at 15 liters, and then, as needed, substitute like-for-like purification resins and membranes, all post-approval. Our bioreactors have a working volume from 2,000 to 6,000 liters, so this dynamic range is somewhat unique in the industry, I think. We always encourage engineering runs, as well as at least 3 PPQ batches, which of course is standard, if not more. And what's cut off down here at the bottom is, you can see this is for a fed batch process. For clinical yields, it's hundreds of grams, and for commercial, we're at 18 kilograms per batch. The 200 to 500 liter scale up, the intent was to maintain the titer, which was roughly 2.5 grams per liter. We conducted several analytical tests. You see here representative testing for size exclusion for aggregates and monomers, CE for main peak, as well as acidic and basic peaks, and of course the glycan profiles. So the key message here is that analytical comparability was spot on from 200 to 500 liters. Now, the more challenging aspect, of course, is scaling up, because now we have to consider the cell culture parameters. So viable cell density and viability were all maintained. You'll see here on the right, comparing to 500 liters, scaling up to 6,000 liters actually allowed us to increase the titer just by changing cell culture parameters. So we are now close to roughly 5.5, 6 grams per liter. And that's why we can get 18 kilograms per batch. Protein purity remains the same. Acidic peaks, main peaks for ICIEF are also comparable. So Project B, scope of changes. The mandate was to lower COGS for an already licensed commercial product. We used the same approach, except here, the initial cell culture method was perfusion, so very high titers of close to 10 grams per liter. However, we decided to drive down the COGS. We needed to maybe take a step backwards and go to fed batch, which for 6,000 liters, based again on our platform approach, we are able to maintain a titer close to 5 grams per liter. A slight increase in yield allows us to get roughly 20 kilograms of DS per batch. The same type of analytical comparability as well as cell culture parameter comparisons were performed. You will see here some representative results for size exclusion chromatography for aggregates, the CE SDS for purity, as well as the glycan profiles. Key message, everything the same with a scale up of 12-fold. And finally, Project C, lowering the COGS before entering into phase 3. This is going to be a successful clinical or commercial product in the eyes of our client. You will see here that the overall yield scaling up from 200 to 6,000 liters is roughly decreased to 50%, but there is a corresponding increase in titer just by optimizing the upstream cell culture from 5.5 to close to 8 grams per liter. And I will close by presenting the comparability data for this project. Again, everything looks good from a cell culture perspective as well as a product characterization perspective. I do have supplementary materials, but I'm out of time, so I would be happy to answer any questions. Thank you for your attention.
Max, that was a very good presentation. I would like to come back maybe on case study number 2, and I would have more of a generic question on this one. So for a lot of projects, it's highly desirable to drive down the cost of goods, okay?
Yes.
So first, I would like you to educate me on this one. That's a very naive question. I would like to understand if there are some materials, raw materials, consumables that we can find only on the Chinese market. And somehow I would like to understand what are the tricks that you're using to drive down the cost of the goods.
That's a very good question, Nick. So there are 3 areas of raw materials that make up more than 80% of the cost of goods. And that is the cost of resin, the cost of filters and membranes, so these 2 materials are reusables, and the cost of cell culture media. Now, we're agnostic. We depend on the clients to tell us whether they want the so-called gold standard, the well-known resins and filters and membranes from Millipore, from Thermo, for example. So we do not control that kind of pricing, except to work with the local distributors to give us the best prices for these overseas imported materials. Where we save a lot of cost is if the client is open to it, employing our own in-house cell culture media and optimizing the upstream cell culture process. You will see on this slide that there are 3 phases, if you will, of reducing the cost of raw materials. The most superficial phase is just a strategic cooperation. We sign some sort of agreement with the distributor or the vendor. The second part is co-development, where we share the risks of perhaps developing new modalities and new applications. And finally, in-house self-development. So there are many CDMOs in China that are developing their own cell culture media. And that, in conjunction with their own self-developed high productivity cell lines, drives a majority of costs down. Aside from that, I think that there is an untapped area where we have a combination of process improvements like continuous purification, as well as employment of new types of bioreactors and perhaps new types of purification resins that can drive the cost of goods even lower. And that's why I was very excited hearing about these new technologies yesterday. So that's about it. Does that answer your question?
Thank you. Yeah. Thanks a lot.
Thanks, Max. I have a question about the same case study, the number B, I think it is. There you change from perfusion to fed batch. That's quite uncommon to change the production system. Is that just to fit to your current facility, or is there another reason behind it?
Well, the main driver was to reduce COGS. Perfusion is somewhat expensive still. And there was agreement with the client that the best way to lower COGS was to go to a more traditional fed batch process. So it is somewhat of a step backward, but with our current needs, again, per batch yields of about 20 kilograms is not too bad for 6,000 liters. We felt that this was the best solution to take a step backward, if you will, from perfusion to fed batch.
In my experience, it depends on the titer of the perfusion
Yes.
setup, of course. That if that's in the range of 4, 5, 6 grams per liter per day, then it's very competitive to the fed batch situation, of course. And the other question I have is on the comparison you made between China and working in China and working internationally. You sort of sketch it as in China there's quite a lot of disadvantages compared to international in what the government demands from, let's say, companies in China. Doesn't it have a big upside if you want to export your products to abroad, the way you're forced to do it?
Precisely.
Okay.
and that's why I'm exploring the opportunity to explore DS at minimum to low and middle income countries and emerging markets. Because we are forced by the environment to lower the cost of goods. There's no question. And the Chinese market is not big enough with the capacity that we have to run at 100% capacity. So we have to go overseas, and it just so happens that we already have developed the efficiencies and implemented the efficiencies to be able to, I'm going to show a slide which I didn't show. To drive the cost of DS per gram to, I show here 106 euros per gram. We can do it now at roughly 70, 80 euros per gram. Meaning each 300 milligram vial of a vedolizumab biosimilar, the COGS is 30 euro. Think about that, 30 euro per vial.
Lower is my experience. Okay.
Yeah. Yes. If no further questions, and if questions further arise, I'm sure we can discuss it later. So thank you for your attention. I think now we have a short break, 30 minutes. We will reconvene here at 10:40. Is that right, Una? Oh, sorry. 11:10. Okay. Feeling the effects of jet lag. You'll have to pardon me. Enjoy the break, everyone. Thank you. Shall we get started? Our next session is called Strategic Evolution in Biomanufacturing: Formulation, Risk Mitigation, Facility Design, and Technology Trends. And our first speaker, I believe, is going to be continuing where Nick left off earlier. Richard Liu is a senior scientist at UCB Pharma, and he will be presenting challenges in adeno-associated virus downstream process and viral safety. Richard, please.
Hello. Hey. Hi guys. My name is Richard Liu. I'm from UCB Pharma, the same company as Nick. So it's my pleasure to present challenge in recombinant AAV downstream process and viral safety. So I cannot avoid this because I need to introduce my company first. But I think Nick had a great introduction already. So our company UCB contribute a lot for R&D. Over 30% of the revenue to contribute to R&D. So that's why I'm here. They recruit many international employees like me, and it's also my pleasure to work in UCB and with Nick. So today I will focus on two topics. First topic, I will dive into maybe a little bit of detail for the recombinant AAV downstream, and second one I will talk about some details about the recombinant AAV downstream viral occurrence study and viral safety. But I think this topic also, very hot topic in the protein biology, like mAb and fusion protein. So I think we can have a great discussion. So firstly, I will dive into what's a challenge in recombinant AAV downstream process, and maybe we can do some comparison with protein biology. So first the slide, I would like to introduce a little bit about what's a recombinant AAV and what's a gene therapy. So the AAV means adeno-associated virus. It was discovered in 1965 and is a part of the parvovirus family. So it's a non-envelope virus, very small size, like maybe less than 35 nanometer size. And this virus has an advantage for human. It's unable to infect a human. So that's why, this AAV can be used by the viral vector, can treat the patient. And why we call this recombinant AAV, because, we just replace the AAV genome to our interest genome. So we can, the AAV can bring this genome into the patient, maybe specific organ. So replace, the dysfunction genome so that then we can treat our patient. So that's why we call recombinant AAV. So compared to protein biology, recombinant AAV is more complex. But for mAb and maybe fusion protein, we just handle the protein. But for recombinant AAV, we have maybe 60 different units, composed of the capsid, and we have interest genome inside. So for here, sorry. Okay, but let's see my red. So for the purification of our AAV, the most critical important issue we need to face is we need to separate this recombinant AAV. So we use our host cell to generate our recombinant AAV. But in the host cell, the recombinant AAV can be assembled to maybe at least we may have these several different types. Maybe this we call this a 4 capsid AAV, means this AAV is a company type. So we have the 4 capsid and we have the 4 genome inside. And the middle one is empty. So empty means, when the assembly process, in the middle of the assembly, the capsid protein can assemble to the capsid, but they cannot assemble with the interest genome. So we need to remove this during our purification, and we keep this one. And also we have the other impurity, the fragment of the DNA. I think Nick had a great introduction earlier. And we maybe need to purify maybe the subunit fragment. So compared to protein biology, for recombinant AAV downstream, we need to face a low yield because we need to separate this one, and this one. So I just put a picture here. Okay, maybe post harvest, we have this capsid, we have empathy, we have many different fragments. And after post clarification, post chromatography, okay, we can get the full capsid, but also we remove lots of this empathy capsid or fragment. So back to the point I showed below, okay. During the caps package, okay, we can get the full empty impurity, okay, maybe the other type, blah, blah, blah. But now, even we have no idea, okay, what's exactly, what kind of different types of capsid we can get. But the full capsid is the only one we need. And so, another issue is, for empty capsid and the full capsid, okay, the two capsid, they have the same subunit. So their isoelectric point, okay, are very similar. So typically the full capsid, their isoelectric point may be 5.9, and this one maybe 6.1, whatever, very close to each other. So even we use some, ion exchange column, okay, it's difficult to separate the two. So that's why, when we purify the full capsid, we need to handle and think about how to overcome the low yield and how to efficiently separate the full capsid, empty capsid, and the other impurity. So let me dive into what's my main topic today. So for recombinant AAV downstream key bottleneck, for now I just organized the 4 points here. So we need to, like I said, we need to face the yield loss. And we have empty partial capsid to remove, okay? And we have a key step, okay? We call that a full enrichment, because in that step, we need to get as much as a full capsid as we can. And the third point is, during the development, we need to provide, drug substance or some in-process material for the other purpose, okay, for maybe the analytical method development or the other, clinical demand or tox demand, okay. But for recombinant AAV compared to protein biology, our yield is lower. So we need to think about how to overcome this bottleneck. And the final bottleneck is, like I mentioned, okay, not only empty partial capsid, we have the other impurity, okay, process product related impurity, and all the other contaminant to remove. So let's talk about the first one, yield loss. So for typical recombinant AAV downstream process, okay, we will try our best to reduce the unit operation as many as we can. So typically, we will do like a 2-column process compared to protein biology. Okay, typically MAB or fusion protein, we have a 3-column. But for recombinant AAV, we typically have 2 columns. So one affinity, another one is IEX. And then we have a VIF, and then of course we have a UFDF to do the buffer exchange and concentration. So, and typically after clarification, we can see here, we will just directly load the clarified harvest into the affinity column. Okay, we may not be able to do additional, buffer exchange to save the yield. And second one, like I said, we will try to reduce the number of downstream steps. And typically we'll follow the 2 chromatography step purification process. And we will try our best to shorten the process time. So for example, we can try to shorten our loading time or process time for the UFDF to save our yield. And the second point, like I mentioned, the empty partial capsid removal is a very critical point for recombinant AAV downstream purification. So typically we'll choose, our polished chromatography, ion exchange, to separate our empty and full capsid, and to separate by different isoelectron point. And we also, have our critical test item. For example, we'll test the viral genome titer, particle titer, and purity, like we just test how much percentage of the full capsid we can get, and we test the DNA content, HCP content aggregation, like a map. So here is our case study, how we implement our IEX to generate more, full capacity. But we may compromise, our yield. So we can adjust our EQ pH. Typically, if we lower our equilibration pH, okay, our percent flow will increase. So we get more full capacity, but our yield will be lower. But I think that makes sense to everybody because we maybe remove more empty capacity or partial capacity. But since empty capacity, they have the similar isoelectric point as the full capacity, so we will also co-elute some full capacity. So the yield will be down. Yeah, but I mean this one just give us some hint. Okay, maybe we can base on different during the development different drug substance or different material need. Okay, maybe we can choose this one condition. Okay, maybe we can have a more higher yield, generate more. Maybe the quality not so good, but we can still provide the material for early phase development need. And if we need for the maybe late stage, we need a very very pure product, then maybe we can go forward this process. So it's a kind of a process adjustment strategy for IEX. So this one just follow up what I mentioned. So how to overcome during development any drug substance or in-process material need. So I think everybody know. Okay, during the process development, okay, even in early stage we have R&D. Our analytical teams need some material to do the method development. We have our own method development need, and we have asset development of all this activity. We need material to do the study. And for TOS, scale-up study, stability study, assay qualification, blah, 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 and late stage process characterization, assay, our analytical team may need a standard for the assay development, viral current study, viral current validation, blah, blah, blah. All these activity, we need the material. So for downstream, we need to generate some useful strategy to generate more material for this need. So for now, for recombinant AAV downstream strategy, okay, we try our best to develop a platform. Okay, the platform should be easy to suitable for different serotype of AAV. But now we have 9 different AAV type in this world. AAV 1, 2, 3, 4 to 9. So different AAV may have different performance, and also we need to make sure our purification step can be scaled up. Okay, because typically AAV is a new area, so most study in the early stage, they develop the AAV in the lab. So they use, ultrafiltration or something else, that not easy to be used in manufacturing. So for now, if we do the CMC process development, we can need to develop our purification process can be scaled up. Okay, for example, we need to develop to membrane filtration and the column chromatography process. And we need to make our process adaptable for any different situation, like I mentioned. Okay, we need to develop our affinity process can be high yield oriented, and our polish process can depend. Okay, if we need a high yield, we can go yield oriented. If we need the product quality, we can go enrich oriented. And our UFDF process need to go for high concentration dosage, because currently the trending is we based on our clinical need, our dosage will be as high as they got, like a map. And 10 years ago, our map maybe dose like a 40, 60. Now most of the dosage maybe like over 100, and predictable. So for now, we are working on like we discussed yesterday, digitalization. We try to analyze our history data so we can do some modeling prediction in the future. And for the final point I want to mention, so for recombinant AAV downstream process, not only empty partial capacity, we have the other impurity to handle. So I joined many discussions with the Bioforum, yeah, and this is survey what I got. So most people from the other company also agree. Okay, for recombinant AAV downstream, we have host cell DNA to handle, like Nick mentioned earlier. And host cell DNA, we have different type, in capacity or outside of capacity. Okay, I shall show here. And we need to think about how to handle the plasmid DNA, because for current general host cell expression system for recombinant AAV, we may need to transfer maybe 3 or even 4 different plasmid. So the plasmid DNA impurity might be a kind of an issue also. So for the host cell DNA, I think since Nick has mentioned this very deeply, so I just do another introduction here. So for host cell DNA, for now we need to follow a guideline. I think similar to MAB and fusion protein. Okay, we need to follow limitation content maybe less than 10 nanogram per dose. Okay, we need to limit the size of our residue DNA can less than 200 base pair. Yeah, and I just show a picture here. So majorly the impurity for recombinant AAV downstream. Okay, we have this one is what we need for capsid, and we got empty, we got partial. Partial means we got capsid, but only not complete intrinsic genome inside. And the DNA, the red one, okay, maybe inside the capsid, okay, and maybe outside the capsid, okay, or maybe inside capsid, but this capsid is empty. So we have many different impurity types to handle. Okay, so for downstream, we also have our strategy to handle this impurity. So for example, for the DNA outside of capsid, okay, we can try to like a traditional chromatography watch step. For our affinity, we can add watch 1, 2, 3, 4. Okay, can adjust different salt concentration, pH. Okay, add some the other additive. Again, we can also try endonuclease addition. For now, we have several article published. If they treat endonuclease into the like affinity or even AES, okay, this endonuclease help to damage or clean this DNA outside of capsid. And for this one, DNA inside the capsid is a big issue. Okay, because for downstream we are difficult to clean them, because if we need to clean them, we also need to clean our capsid. So for this one, we can try the other way. Okay, but I think this is not only the issue for us, for the other AAV development company too. So we will try to remove more empty partial capsid. Yeah, because like I mentioned, this DNA may be inside the empty partial capsid too. So we can is several reference. Okay, I show here. They said they can try some additive like a histidine or detergent like a P1A or PS20, PS80, and they're also helpful to help to remove more empty partial. But the side effect is like I said, when we remove the empty partial capsid, we also remove some full capsid because their PI is very close. So for this way, it's still a very huge challenge for recombinant AAV downstream, because this way, okay, like I said, we just treat the host cell as a product-related impurity. So when we clean this, we also impact to our process yield. Okay, so this is a final summary slide. So I just summarize the 4, for now is a huge bottleneck for recombinant AAV downstream process. So we need to face yield loss. We need to try our best to remove the empty partial capsid. And we need to think about how to, have some generate as much as material to our, for our different need for analytical team or for our the other development need. And finally, we need to think about how to handle the other process and product-related impurity contaminant. Okay, so for yield loss, like I mentioned, try our best to reduce the number of downstream unit operation. And for empty partial capsid, okay, like we can, do some adjustment from the process parameter perspective. For example, the equilibration, pH. Also, we can, think about, to adjust the other process parameter too. And for the DS or in process demand, okay, we try our best to develop our platform, okay, scalable, adaptable, predictable. And for the other impurity, okay, for example, for the DNA outside of capsid, inside capsid, okay, we have different strategy to handle them. Okay, so the second point I want to mention here, because it's another hot topic for gene therapy, especially for recombinant AAV, is about viral safety consideration. For recombinant AAV, we can leverage some experiment from protein biology, like from MAB and fusion protein, because for recombinant AAV, our purification step is very similar, but just maybe less unit operation number. So I think the viral safety is very important, not only for recombinant AAV, but also for all product. Yeah, I think FDA very focused on this. So this the whole, overview for the biomanufacturing process. So we have starting material, MCB, WCB upstream, harvest, EOBC downstream, DS. Okay, so for every different stage, we need to, have different strategy to do the viral safety control. For example, we need to do the viral testing for starting material, MCB, WC, blah, blah, blah. For harvest, we need to have the UP, unprocessed bulk testing to make sure we have no viral contamination. And we have ICH Q5A guideline. Yeah, this is one, I think the guideline is very famous. So this guideline, especially to guide, how to do the viral clearance, especially for late stage for manufacturing. Yeah, so I just list, the 7 critical points here. I think everybody should be familiar with that. So for example, we need to have an orthogonal mechanism, at least the process step for, can be evaluate the viral clearance. And we need to distinguish between the viral removal inactivation, blah, blah, blah. But for recombinant AAV, compared to protein biology, we have some gap. For example, like I mentioned, okay, our purification step is less than protein biology. So we may not have sufficient unit operation, can be evaluate viral clearance. And also our model virus will be different. I think that makes sense. Maybe we use the different host cell and the different possible contaminant viral source. If we compare recombinant AAV and the protein biology process. And for now, since the gene therapy product is very, very diverse, okay, maybe some product, their downstream process is only one unit operation, like UFDF, they just need to do a buffer exchange, for example, for cell therapy. So there's no mandatory to say, okay, especially for early phase, for gene therapy or cell therapy, we need to do the viral clearance study. But it didn't say you don't need to do. It just say you may need to do some risk assessment. Okay, if you can justify, okay, your product is viral safety is okay, then okay, you don't need to do. But if you after you do the self-assessment, you think you need to do some viral clearance study to prove your biosafety, then you need to do. So we had lots of discussion with our bioforum colleague. Okay. More than 60%, okay, our colleagues, they say the project will go forward to do the early phase viral clearance study, which means, okay, even for gene therapy or also cell therapy, we need to make sure our viral safety. Yeah. And also, they say if we can do the viral clearance study as early as we can accumulate some live virus clearance data and prepare for the late stage. So, like I mentioned, for recombinant AAV downstream, okay, we have different model virus. Okay, so here is like we also done some survey in bioforum. Okay, this is from maybe FDA or in the institution from 2023, 2024. The major model virus we choose for recombinant AAV process. So we have ADV5, SV40. I show here. MOV, MVN, similar to MAP and the protein biology area, and this one. So, but for model virus selection, I would say the strategy is similar to protein biology. We need to base on our, host cells system, transfection system, something like that. And for the downstream, we can leverage some experience from the protein biology. So for protein biology, like I mentioned, okay, we have many steps to do the purification, like affinity, low pH inactivation, AES flow through, and AD flow through, and the nanofiltration is a robust viral removal step. But for recombinant AAV downstream, okay, we have less number of downstream steps, so we can consider, okay, before the affinity, okay, we can do some detergent inactivation, and we have affinity to do viral removal step and the AES to do the viral removal step. But this AES may not be like a MAP or fusion protein. It's a flow-through mode, it's a binding mode, so it may not be the robust. And for the nanofiltration, because our AAV particle size is larger than MAP and fusion protein, so the pore size is more than 35 nanometer. So maybe just certain size of viral can be removable. So we also did the survey with a bioforum colleague. Okay, they agree from the 2023 to 2024, the detergent inactivation is more critical because more clients choose to do the data-driven activation at this step into the viral current study. So for now, the RAV viral current strategy will aid, most people will agree to aid the data-driven activation into the viral current study. So I just make a summary table. So for recombinant AAV viral current study, we need to consider data-driven activation and consider our 2 chromatography step and the viral infiltration. And for the chromatography step, because our VIF, okay, our process is larger than compared to a mAb and fission protein, so maybe just we cannot filter the small size of virus during the step. So we need to make sure our chromatography can clean those small size of virus. So this is another case study. My time's up, so I try to short. Okay. So this is some data from the very interesting case we discussed in the bioforum. So they just decide the viral current study. They choose the data-driven activation, affinity chromatography, AES, and the viral infiltration. And they can get more than 4 log reduction value from the VRF and the viral inactivation. And chromatography, they can get total reduction value 6 to 7. And they submit to Andy. Andy said, okay, this data looks good, can provide sufficient clearance. But they also get some feedback. Okay, so they need to calculate, for example, under the worst case, okay, worst case viral burden, what's the final, possible infection particle in the DS, and if in the future their drug substance dosage increase, okay, maybe is that possible, can get the risk to get the viral contamination. And also the final one is they also asked the sponsor to calculate, okay, not only virus, but also maybe the other, like a viral DNA or capsid protein fragment to see, okay, if they need to consider this impurity in the drug substance. So this is our final slide. Okay, the summary for the recombinant AAV viral current strategy. Okay, for early phase and late stage. For late stage is easy, we just follow SHQ5. For early stage, just like I said, for now there's no clear guideline, but it depends on our experiment and what I mentioned. Okay, we need to make sure, okay, our process, total step can provide a significant clearance for, the possible contaminated virus. And we need to discuss with the regulator, depends on our product, to do a specific strategy, and also to evaluate the virus, based on our transfection system. So these are my final takeaway. Sorry, I just go forward. So I mentioned several 4 major challenge in recombinant AAV downstream, and I mentioned several, critical point for the AAV downstream viral safety consideration. Sorry, I run out of time. So this is my team. We are a new team UCB from the gene therapy process science. Our head is Kyle, and the upstream head is Nick there, and I'm from the downstream. So this is our team. So thank you guys. Sorry to be out of time. I'm happy to take questions.
We have time for one question.
It was interesting to hear that solvent detergent methods are being used again for viral clearance, because historically they have been used for plasma protein removal of viruses there, but for recombinant proteins they were not used then anymore, and now it's coming again back. Do you use the same solvent detergents as used for the plasma proteins, or are there different ones?
So your question is, what kind of detergent we will use for inactivation? It depends. We may use like a famous one, PSAD, PS20, TMBP. You can find this from the article too. But recently, I think in Europe, we also consider Triton S100, but in Europe it's banned. But in US, not banned. So we are looking for something can replace the Triton S100. But what I heard, most people will use PSAD, PS20, and the TMBP. So, you can just combine 1 or 2 or 3, and yeah. So, and you can, refer to the article in the Word. They suggest several useful concentration for different, PS80, PS20. Yeah.
Thanks.
Yep.
Thank you, Richard. Our next speaker is Maya Leiven from Lenio Bio. She will be presenting next generation bioprocessing technologies, trends, and innovations. Maya.
Thank you. So I'm going to give you an overview of our technology and how we think it can revolutionize the biomanufacturing processes. It's an efficient and scalable eukaryotic cell-free protein production platform. I included a short introduction of myself. I have a PhD in synthetic biology and bioengineering from the Technical University of Denmark, where I mainly focused on the development of synthetic biology tools for protein production optimization. So I'm more of an upstream person, not so much downstream. I then founded a spin-out from the university and was for 3 years CTO of that biotech startup that offered a B2B service for prokaryotic protein production optimization. And since then, I'm with Lenio Bio, the company that I'm also presenting here, heading the protein solutions department, where we are enabling production of difficult protein targets for our customers. So all in all, I have 10 years of experience in protein production and the protein production optimization field. I think we are in a very interesting time at the moment. There are more and more new and very complex modalities coming up, but those are also challenging the existing development workflows. So I try to illustrate that here with this graph. So you have the time and resources that are required for development, and then you have the complexity of biologics. And you can see that this curve is like rising up with more and more complex modalities, like, for example, multispecific antibodies. But also yesterday we, for example, talked about AI-generated molecules. And cell-based protein expression technologies often struggle with long lead times and low yields and slow iteration time for those new modalities. And we therefore see the need for new technologies that are required to flatten this curve here. And the solution that I want to present to you is ALICE, which is a plant-based, fully scalable eukaryotic cell-free protein expression platform. Why cell-free? So cell-free protein production is fast. It can express in days versus weeks or months for cell-based systems. It's very versatile, capable of expressing diverse classes of proteins, those that require also disulfide bonds and glycosylation, so cross-translational modifications. And it's very low touch, meaning that no transfection of a cell line is required or no cell line engineering, and there's a minimal hands-on time to use a system. So cell-free protein production has been around for a very long time. However, traditionally cell-free protein production has failed in being scalable. So it was more used for very small research-grade reactions. So with our system now, scalability of cell-free protein production is possible, and then can actually reduce the time for drug development and accelerate the speed to commercialization. So in a cell-based expression system for like very complex modalities, cell line development can take up to a year and more weeks than for production of a protein. And in ALICE, there is no cell line development required. You can produce a protein within 2 days, and downstream purification methods are same as for cell-based systems, so nothing needs to be changed there. So our self-free platform enables you to test more, to fail early, and to adapt fast. I'm going to tell you a little bit about our self-free protein expression system and how self-free protein expression in general works. So you have basically two processes. So in the first process, you require cell culture, right? So somehow you need to produce your cell-free extract for protein production. In that lysate production process, we take a plant cell line BY2, which is from Nicotiana tabacum. We grow the cells, and then in the lysate production process, we remove everything from that cell that we do not want or require for protein production, and we concentrate everything that is needed and beneficial for protein production. Then this lysate gets also mixed with some optimized master mix components that are required to enable a successful protein production reaction. And at that point, we can freeze the lysate and then take it out on demand, just add the DNA and produce a protein. Here I've highlighted some classes of proteins that we can very successfully produce in our ELIS system. For example, antigens, virus-like particles, also small peptides, antibodies, multispecific antibodies, growth factors, cytokines, and a lot of enzymes. Also membrane proteins, for example, very large proteins. So why is it possible to produce so versatile proteins in the ELIS system? When you look at how the ELIS system is used, I told you that it is really only DNA that needs to be added, so everything else is already in there. That means, like in a standard cell-free system, reactions are very complex. A lot of different components need to be added to the reaction. We have already incorporated all of this with our master mix that we add here, and have optimized that master mix so that you get an agnostic lysate that really works for everything. And in the process, so that is just highlighting again the different classes of proteins that I already mentioned to you. So when you add the DNA, you have two different options in the system. And that really comes with the process and how we produce the lysate. So in this production process, we keep so-called microsomes in the reaction. So microsomes are consisting of ER and Golgi from the cell, which is the compartment where post-translational modification is performed, like disulfide bond formation and glycosylation and such. And then we have two different vectors that are used in our system. The PLS1 vector is guiding the protein into the cytosol of the lysate, where complex proteins can be produced that do not require post-translational modifications. And then we have those so-called microsomes, where the PLS2 vector is guiding the protein, aided by a signal peptide sequence. Similar to a cell-based system, the protein gets then imported via the sec-translocon and gets post-translationally modified. Those microsomes also give the chance for membrane proteins to be natively folded in the cell-free lysate. So other cell-free lysates do not come with inherent membrane compartments in there. So you need detergents, nanodiscs, some aid for a membrane protein to fold correctly in a cell-free reaction, whereas here the protein can just insert into the membrane of the microsome. Compared to other cell-free systems that are available, our yield is much higher. And due to those microsomes being in the lysate, we are also capable of producing proteins that require post-translational modifications. However, cell-free production is in general, even when I'm stating here that our expression technology is rather low price, cell-free expression is in general still a very cost-expensive field. We also have some external support, for example, a study that was performed by Sartorius stating that our system is outperforming other cell-free production systems, so it's not just me making up those numbers here, they have also been tested. So now I want to venture into some case studies to show you how actually Alice can reduce the development timelines for complex proteins, and also look a bit into the quality of the proteins that we get in our system. In September 22, there was a global shortage of the enzyme alteplase. Not sure if anyone has heard of that, but it was widely covered in the media that this enzyme was used for acute ischemic strokes could not be produced. It's like a medicine that is launched by Boehringer Ingelheim and Genentech, and it transforms plasminogen into plasmin, which then starts the removal of blood clots. And that is, of course, a medication that in nowadays, where diseases in that direction are very common, a medication that is very commonly used and also required. And the manufacturing process was cited as the main contributor for the shortage of that enzyme, because there have been challenges. It's a very complex enzyme that has a lot of disulfide bonds, glycosylation sites, those so-called Kringle domains. It also has here a cleavage site where an autocleavage of a protease domain needs to happen. And it is just very complex, so that it cannot be easily adapted to produce that protein without having access to the proprietary cell lines that are used for that. So we decided, actually, let's just order the DNA, put it into our lysate that we took out of the freezer, produce the protein, first in a small scale to test, can we actually do that? That took us 2 days. Yes, we were able to produce it. We kind of calculated, okay, how much protein do we want to have? Let's start a 20 milliliter reaction, took out the lysate of the freezer again, started the reaction to purify about 1 milligram of that protein, and then test that in activity assays to see if we would actually get the same quality of material as was obtained when producing it in CHO cells, as it is done commercially. And when performing an activity assay, you can actually see that our product, the red line here, was actually even showing a stronger activity than the CHO reference that we used. So yes, we can produce high quantity of very high quality protein. We also ventured into comparing our Alice-produced proteins to other commercially available products. In this case, that is like a very well-known pharmaceutical, Humira, also called adalimumab, an antibody that is also produced in CHO cells. So we produced adalimumab in our cell-free system. We saw batch-to-batch production consistency. We saw that all disulfide bonds were formed. The thermostability of Humira and our Alice-produced adalimumab were similar. The identity was also confirmed, and they were also comparable target engagement and Fc receptor binding. We also performed a single-dose pharmacokinetic study, and those showed comparability in half-life as well as Tmax and Cmax. And our Alice-produced adalimumab might even have performed a little bit better, but that is a one-time study, but we are very proud of it and think that there's a bright future ahead for our system. So enabling production of proteins is great, right? But when you then can finally produce a protein, where does it bring you when you cannot scale it up, right? There needs to be scalability to move further into manufacturing. So that is why we have ventured into scaling our protein expression. We have received funding from the European Innovation Council for doing that, and we have built a pilot plant for our lysate production process, where we can produce lysate from 500-liter cell culture, which results in 10 liters of lysates per run. Yeah, so those are real pictures from our pilot plant facility. And after we had scaled the lysate production process, we of course also needed to scale the protein production process with having then lots of lysate on hand. And also here we increased from our standard reaction scale of 50 microliters to a 10-liter reaction in a shaken bioreactor. And I have brought here an example for a microsomally expressed protein, and you can see that across scales from 100 microliter to 100 milliliter, you can see linear scalability of the process. And we have also gained a lot of expertise in process development in that process, and are really open to also support the adaptation to other reaction vessels that we haven't yet used. So for cell-free systems, at least for ours, because we have active mitochondria in the system, oxygen demand, for example, is a very important topic. So expertise is required to tune the process to make it work in any reaction vessel. The third case study that I brought is a case study about virus-like particles, VLPs, which are used for vaccines that can be decorated with antigens. And we use our system to first, in small scale, screen different candidates and select them, and then scale the reaction to actually then do immunological testing. Here we have an example that we have published about scaled VLP production in Alice, and you can also see here that over a large range of reaction volumes, we actually saw pretty high titers of very uniform VLP particles that were also immunogenic. So they were recognized and endocytosed by by IDCs, the fluorescently labeled VLP particles you can see here. And they also showed cytokine response of PBMCs. I'm running out of time. So what I also wanted to show you is that we are actually supported by CEPI. We are supporting CEPI's 100-day mission for pandemic preparedness. The goal is really to compress the timeline to develop safe, effective, and globally accessible vaccines in just 100 days. That is really the time span where, for example, the COVID pandemic could have been contained. And we are about to show that our cell-free technology can be very well used for that. Our roadmap, if there is interest in using the Alice platform, we have a research kit and also scale-up material that we are selling via our web shop and also via other platforms. We have a service that we have launched 4 years ago. We have a lot of experience in protein production. This department is headed by myself. We have a path forward to guide a project through different process steps, feasibility, proof of concept, scaling, and eventually also tech transfer for seamless in-house protein production with our system. And we are also in the process of implementing Alice for high throughput expression, and we are having a path towards GMP protein production. I want to give you a few minutes for questions, so with that, I want to thank you for your attention. As a summary here, the final takeaways.
You have time, Maya.
I have time, and
Yeah.
it says 1 minute
Yeah.
only.
No, go ahead.
Yeah. So yeah, just summing up, we have scaled the technology. We have shown that that cell-free technologies can be scaled in a range of volumes and reactions vessels. That is the first time ever that was shown for a eukaryotic cell-free expression system. We can produce highly complex and functional therapeutic proteins without specific optimization to the system in just 24 hours. And yeah, we are well on track to deliver a transferable licensed manufacturing process to any customer to support large-scale protein manufacture. And we also are on track to full GMP compliance, enabling future clinical trial support of proteins. That's our scientific team. And of course, I have to thank all of our funding and you for your attention.
We have time for several questions. Nick?
Maya, I'm really not happy with you. I like cells, and you're trying to kill my business here. Besides that, what I wanted to say is I have difficulties to understand the economic behind the system that you're developing, because it's not synthetic production. It's an intermediate in between a cell and a cell lysate, and then to produce let's say 10 liters of starting material, you need 500 liters of cells, for which you also need reagent and so on. So I would like to understand better how this can compete with cells that are professional at producing bioproteins. Could you help me to understand that a little further?
Yeah, I think it's, I mean, the economics of cell-free systems are at that moment not great yet. That is definitely the case. But however, they are enabling things, right? So it's also often a question of what do I want to produce, or what do I need to produce? How efficient does that process need to be? And that is where cell-free has a big advantage. It's not a living system. It can perform everything that a cell can do, but it's not a living system. That means cytotoxicity, for example, is not a problem. There's no host cell DNA in the system. There are no viruses in the system, for example. For especially you, that might be an advantage. AI-generated molecules, there needs to be rapid testing of those, right? So AI generation will stop at some point if there's like no platform to test the molecules. Very often, that takes too much time in a cell-based system, or the molecules are just too complex. So that is really the unique points where cell-free comes in. And we need to get early adapters to further then develop the technology and make it at some point then economically viable. Yeah.
Thank you, Maya.
My question would be, why actually tobacco? Because and versus CHO cells or HEC or whatever other cell lines. Is it that the, I don't know, ribosomes are more stable or so? And the second question would be, what do you run out of then when the reaction goes down? Is it that you run out of tRNA, or I don't know?
Yeah, I think first of all, there is a CHO cell-free system. People are trying to develop a CHO cell-free system, but the titers are extremely low and it is not scalable at that moment. Also, cultivation of plant cells is pretty cheap. So maybe also coming back to the cost of goods and the economics behind it, right? Like running a 500-liter CHO cell fermentation to then just not really use it for any production, but just to make the lysate is really, really expensive. So plant cells have a really cheap media composition. So the cost of goods for running a fermentation with plant cells is very low. And then, yeah, it must be something about the robustness. I mean, we're not 100% sure. It's not really, we are happy that our system is working, right? So we haven't really investigated why that is the case. But yeah, it is a very robust system. So we are able to achieve pretty high titers and scale it. And that is currently not possible for other eukaryotic systems. You also had a second question, right? I only answered one.
What you run out of at the end of the reaction?
Yeah, exactly. So I think it's mainly, like you said, nucleotides, amino acids, things like that. So you can also do continuous cell-free reactions by having feeding, what we are currently not doing. Then can of course prolong the reaction, also increase titers, but it also raises the costs, right?
Okay, hello, thank you for the presentation. I have two questions. The first one is related to the scale. So what is the biggest reaction that you have, the volume of reaction that you have done with the lysate, I mean, after you...
Yeah, so at the moment it's 10 liter.
And what is why is it limited to 10 liters? Is the previous part that limits that, or is it something related to either oxygenation or shear stress?
No, there is. It would be very well further scalable. Like, we are pretty certain of that. The only thing is that our current pilot plant that produces the lysate produces 10 liter in one batch. So that is how far we have scaled it, but there's nothing that prohibits the system from being scaled even further. Yeah. So if you would, in our current process, pool several batches of lysate that are produced, it would be possible to scale it up to 100 liter or even further, I think.
Thank you.
So Maya, with your case studies, at 10 liters, how many grams of protein? What's the mass that's produced?
Yeah, so I mean, that is still very dependent on the protein. So for our reference proteins that we used for batch releases, we produced 2 grams per liter. But for antibodies, for example, we are at this moment more in the range of 200 micrograms per milliliter. So that's 200 milligrams per liter. Yeah. But as I said, at this moment, it's an agnostic system. So we are more focusing on broadly being able to produce a lot and focusing on like the capability of screening a lot. But it's an open system. That is the beauty of cell-free. You can just add whatever you want. So there are definitive ways to boost expression by making the lysate not agnostic but more specific for a certain protein compound.
One question from Ard. Nick. Okay.
Make sure that the initial question that was asked here is answered. Do you see limitations due to mass transfer, so gassing or shear, sensitivity to shear? Is it something that you're observing when you go with high agitation or?
Not in the general operating parameters, it's a problem. I mean, shear stress, of course, the microsomes are membrane compartments that can break at some point, but not under standard operating conditions. That is not a problem. The system needs a lot of oxygen. So when, like, let's say, the reaction vessel is filled to top with lysate, then oxygenation is required. But yeah, it really depends also on like what reaction vessel is used and how you fill it. yeah. But we haven't seen we haven't seen a case where we couldn't fix the parameters so that the system was working.
I have a final. I'm still allowed to ask a question, or it's okay?
Yes, please.
What are the patented parts of the whole Alice concept?
The cell line is proprietary, for example.
The lysate.
That is used for the lysate. Also parts of the lysate production process. And also the parts of the vectors that are used. So they are very specific for the system. It's a transcription is based on T7, but translation is then based on the inherent plant system. So we have a prokaryotic transcription system with a eukaryotic translation system, basically.
And how long do you? What's your prediction? How long will it take before you can make formal, say, GP products? What's the expectation?
So we have a full roadmap for that in place. We are also, for example, regularly discussing with authorities about how our product is regulatorily seen. Currently it is classified as a raw material, which is really nice. But the timeline to really implementing GMP is based on the customers that we are working with. It's a very costly process, so it is not our highest priority at the moment to make everything GMP conform, but we have the path. All the components we are using in the manufacturing process, they are available in GMP grade, so we would be able to very rapidly switch the process into a GMP process. And we have also just a few months ago announced a partnership with a CDMO, so that took in our protein production process into a GMP facility. So since we partnered with a CDMO, the protein production process can already be performed under GMP. The lysate production process is currently only performed in-house, but everything would be ready to transfer it to GMP.
If no further questions, thank you, Maya.
Thank you.
Our next speaker is Alessandro Scandura, who is the Chief Strategy Officer of Vicomtech. He will be presenting, let me read the title here. His presentation is titled, Explore the Cutting Edge Potential. Oh, Martin, okay. I stand corrected. Our next speaker is Martin Prantel, process engineer from VTU Engineering. And his presentation is titled, Energy, Carbon, and Water Flow of a Biopharmaceutical Drug Substance Facility, including Derived Improvement Possibilities. Oh, I'm sorry, it's a joint presentation, Cornelia. With Cornelia Haas, Manufacturing Science and Technology Team Lead, also VTU Engineering.
Yeah, thank you for giving us the opportunity to talk here on our favorite topic, sustainability. So sustainability in biopharmaceutical manufacturing, or generally in manufacturing, has kind of three gear wheels that can be addressed. First, in the process design and in the facility design phases, sustainability matrices can be applied in different stage decisions. Then, when you have a facility operating with legacy products, this is then different how you need to interact. And you can do this on three levels. You first can conduct an energy audit, which is compulsory, where you look at the main consumers and the main producers of energy inside your manufacturing facility. When you combine this and then go more into detail to an equipment-specific level, you can do an energy flow analysis. And then when you address then also financial aspects, so OPEX and CAPEX costs, you end up at a sustainability strategy implementation. And we are going to present to you kind of a mix of these two things for biopharmaceutical manufacturing. Yeah, just you will all probably know biopharmaceutical manufacturing is divided into upstream process and a downstream process. Upstream process produces the cells, and then in the downstream process the protein of interest is purified. And depending on the operation, different types of cleanroom grades are then used for conducting the different operations. And high cleanroom grades are situated in lower cleanroom grades, which are then situated in control areas. And the entire HVAC system, which runs these clean rooms, is one of the major energy consumers in pharmaceutical manufacturing. The second major energy hotspot in pharmaceutical manufacturing is the water and steam generation. So you start out with tap water, purified by ion exchange to get purified water, and then by distillation, at least until 27 at least, to get to water for injection, WFI. And this is a very energy-intensive process, and only WFI can be used for directly late applications with product contact. On the energy side, so for the heat energy that is required inside a biopharmaceutical manufacturing facility, there are different types of steam available, and the black steam or utility-grade steam, this is produced from the tap water by heating it up. But when you want to use it for direct applications, and not just double jacket heatings, you need to produce clean steam where you need to start from purified water, so water that has undergone these ion exchange treatments. We are now presenting data for a model facility for antibody or protein production with 3,000 square meters, so a moderate to maybe a little bit smaller than average size facility, which is based on the data from several facilities where we calculated the energy flows from. This is a stainless steel facility, so it needs extensive sanitization procedures in order to keep the vessels clean. For generating the data, there are lots of flows that need to be mapped and lots of equipment that need to be parameterized and then the data generated from. And the easiest way is if you can do this by physical interlinks, because the input energy must match the output energy. And for typically a clean steam generator, the purified water, the amount is measured, and by this you can calculate the amount of steam that is produced, because you know the temperature and the pressure from it, and from the amount of steam that exits the clean steam generator, you can calculate the amount of black or utility steam that is required to heat up the water in order to produce the clean steam. The next way of getting the data is parameterization of equipment with manufacturing data, so manufacturing data of the equipment manufacturer. This is now shown here for a case of a WFI generator. This type of equipment is an on-off equipment, so it operates either at full blast, so 100% of its capability, or not at all. And then from the process control system, you can derive the operating hours and when it was used throughout the year that is modeled to get kind of to get to the amounts of purified water that are used by these WFI generators throughout the year. And therefore, from This data then, again with the manufacturer's data, the amount of WFI that is produced from it can be calculated, and then going back then also to the amount of black steam or utility grade steam that is necessary to produce it. In the third type of example, this gets even then a bit more complex to get to the actual data. Here for a refrigeration machine, only the amount of electricity that goes into the refrigeration machine is known, and the amount of cooling energy as well as the heat that leaves this heat pump or a refrigeration machine was not known. And it could not be calculated, and therefore we installed sensors, monitored for a couple of weeks, and then correlated the electrical power with the cooling power that was emanating from the system in order to get the balance of the entire system. Yeah, and when you sum this all up for the facility, you get to the energy flows that will be now presented by Martin.
Thank you, Cornelia. Yes, Cornelia already mentioned, we use a lot of data we need to gather from the facility and maybe recalculate whether they are directly available or not. What we get out of that data are energy flow diagrams showing the energy flow per year, which basically show the complex behavior of such a pharma facility in a quite easy, understandable manner. So what we see here, we use quite an extensive amount of energy from electricity. Of course, we need to keep up the clean room conditions, so an extensive amount of energy goes into HVAC systems. Can also be seen here on the cooling energy consumed and on the heating energy. So especially in the heating system, we show like energy flows from different sources like district heating system, from heat pumps, here a small amount, and a large amount, for example, from heat exchangers, which take over steam energy from utility grade steam into heating power. With that extensive amount of data available, we are able not only to show energy flows, but also like operational costs flowing to different devices, and especially interesting now, CO2 flows, which you can see here. That shrinks down the complexity of the diagram pretty much, because as shown here, electricity can easily be bought free of CO2, also like maybe district heating systems. And so here an extensive amount of CO2 comes from utility grade steam, typically seen in farm industries, because the steam is produced by natural gas. Also a very interesting part, of course, is the purification of water in a pharma facility. So there quite a big amount of clean steam or utility grade steam, like around here 13,500 megawatt hours, are used for the heating purposes, as mentioned before. And the two big main energy consumers, the clean steam generators and the WFI generators, for example. We also use an extensive amount of potable water here, which is then purified via purified water generators like membrane filtration and so on, which mainly uses electricity for its operation. This purified water then gets directly consumed in the different production lines for low quality washing cycles and so on. But it also gets consumed by the WFI generators and the clean steam generators. What we already can see here is that the clean steam generator and the WFI generator use nearly the same amount of utility grade steam, but consume different amounts of purified water. Of course, steam creation is a very energy intensive task, so clean steam generator consumes less purified water. We can also see here the CO2 flow of the water purification processes. So here as well, the only CO2 impact comes from the utility grade steam. Of course, clean steam generators and WFI generators so produce the biggest amount of CO2. Yeah, but what we can observe here at the end, at the consumption, at the production lines, that WFI has a really big impact in the CO2 emissions compared to the purified water. As we saw on the page before, there's way more purified water consumed than WFI. So shows already that it really has a big impact whether WFI is needed or used or not. On the overall schematic, we can see different improvement topics. We focus on pharma or in pharma facilities, which can be here on the utility grade steam distribution system. Of course, we have a lot of power and energy flows there. On the WFI generators, and also like on the clean steam generators and the usage in the different production lines. These few points I want to show you more in detail now. Like here on specifically on the WFI distribution system. So typically also in existing facilities, WFI is produced via hot distillation. So it comes out of the generator with around 100 degrees, then stored at a temperature above 80 degrees Celsius. The end user usually consumes this at room temperature, so it constantly needs to be cooled down when it's used. But as we are here in a clean utility system, the water needs to be circulated around in order to operate the clean utility system. For that reason, typically heat exchangers are installed already and additional chillers to cool down the water that comes from the storage vessel. When then it goes to the consumers. When the water comes back, it gets heated up by the heat exchanger first and maybe needs to be heated up again in the storage vessel. What we see here that in typically existing facilities, the heat exchangers are calculated for different points of operation and which are not optimized for energy efficiency overall. So what we usually do, we gather a lot of data, mainly a year round, with 10 seconds, 1 minute data and analyze this if it's feasible to install or it makes sense economically or from the perspective of CO2 to install a larger heat exchanger or not. So like we are analyzing the systems on real world data, maybe with as an add-on, if not enough data is available, we make on-site measurements as well. So in typical projects, we see like with investments here of 50,000 euros, we see operational cost savings in 18,000 euros. So next project which usually saves quite a lot of energy is in the utility grade steam systems. Utility grade steam is used for heating purposes mainly. So like in heat exchangers, the high pressure, high temperature steam gets condensated and gives off energy to the different vessels for heating purposes, for example, then flows through a steam trap into the condensate system. while doing this, it expands from high pressure, high temperature to atmospheric pressure, and for that the condensate gets cooled down and excess steam is created, which then both flow to the bottom of the facility into a collection tank. This vessel is vented above the facilities, where you usually see that common steam pumps on pharma facilities, which basically is waste heat, waste energy, which gets blown into the atmosphere. For that, we created different concepts to reuse that energy wasted on the rooftops. And we see here with investments of around 300,000 euros, like operational cost savings of 50,000 euros, mainly because it could be directly used in humidification systems, so for air humidification, or for heating purposes as well. That's the main use cases we see here. Another improvement project could be the recycling of WFI, because all stainless steel vessels need to be sanitized, of course. And after the sanitizing cycle, the vessels are still hot, and WFI is used for a final rinse. So all the vessel is already clean, and the quality basically shouldn't get worse of the WFI, which is basically wasted. And an additional effect is that the WFI gets heated up to around 80-90 degrees, so we recover as well quite a lot of thermal energy. In the waste systems, we install there a conductivity measurement to see if it meets our targets, and then it could be used for steam generation, for utility-grade steam generation, of course, or it goes into waste if it doesn't meet the criteria. And we see here quite a lot of savings in operational costs, water savings, and CO2 savings are possible there. So these are only a few projects we did. Here we can see other ones, like for example, especially in the HVAC systems. We have large HVAC systems, as mentioned. So in the heat pumps, for example, there is a lot of potential, because also in the regulation of them, because usually cooling power and heating power is used at the same time. So if the systems could match, the heat pump could serve as a cooling machine and as a heat pump at the same time, which needs a bit more to dig into the process and see if it's possible to match that demands. Also, what I want to mention, this is only a combined flow diagram, but basically what we usually do is analyze every different device. So it's quite easy for us to compare different chillers, for example, to each other and see if there is going something wrong on specific chillers. KPIs could be created to match on a yearly basis if there is going something wrong or not. Maybe a valve is closing a little bit more, so the cooling stream isn't the best for specific cooling machines, so which could be seen as well in this Sankey analysis. Also here, additionally on the clean steam generators, we are thinking clean steam as a media for sanitization, but usually it also has a heating purpose of the systems. And the usual stainless steel vessels are equipped with heating jacket as well, and so the heating jacket should be used for heating up the vessels before sanitizing, of course with energy from sustainable sources, and then the clean steam should be introduced to save the expensive clean steam. Concluding, we can say that we with some easy projects in existing facilities, quite a big amount of energy could be saved. Flow diagrams are always a good start for extensive sustainability improvements in such facilities, as a lot of data gets already gathered and which is then available for future projects, and the flow diagrams show complex behavior of these facilities in an easy manner. Of course, in-depth evaluation of the processes is needed in order to get all this data for the flow diagrams, and usual dynamic approaches are needed to recalculate that improvement projects which we want to implement and give a sophisticated answer to the energy improvement to our customers. Yeah, I'm at the end of our presentation and at the end of time, so thank you for your attention. If there are any questions left, we'd be happy to answer.
Thank you for the presentation. I have two questions. The first one is, if you think that this kind of industry is suited for a cogeneration approach. And with that I mean, instead of producing low pressure steam, producing higher pressure steam, using a steam turbine to produce electricity, and then with the low pressure, after that you can use all the low quality energy requirements of the plant. Oil and gas, or even agro-tech of process, combined process, cuts in energy consumption. The second question is, if when you do the analysis of the plant, I've seen that you have been using energy flow diagrams. I was wondering if you also use exergy flow diagrams.
The first question, yeah, of course, like combined steam generation and using the generated steam for electricity production. Of course, people are thinking about that, but actually, I think, especially in paper industry, it's done a lot, like with gas turbines. But now everybody wants to get away of the gas because it's emitting CO2, and also the prices are pretty unpredictable. And so it's more like companies are more going into electrification. So like using heat pumps, maybe recovering the waste heat from sanitization purposes, and with high temperature heat pumps, it's already available. It's already possible to create steam again. So it's more into the electrification purpose because of the emitted CO2.
Mm-hmm. So you're relying on green energy to produce electricity as a initial point.
Of course, yeah.
Okay, thank you.
Yeah. And now we usually use energy flow diagrams, just no exergy flows. Yeah.
Thank you, Cornelia and Martin, for providing focus on a topic that I think not enough attention is paid to. I do have a question, and this is more logistical. So for a full analysis of all utilities in an existing plant, what kind of resources and how much time does it take to prepare for the analysis, number one? And number two, how much time will it actually take to perform the full analysis? This is for a biologics manufacturing facility.
Actually, it's quite a hard question because it's so different from customer to customer, and it's because of the availability of data. And what we usually see, like in the process itself, data is available, of course, but at the utility systems and so on, there are like different systems where you have to combine data, and maybe data is not available at the same level of detail. And so what we usually do is starting off with evaluation projects of seeing which data is available and then giving a concrete time estimate, I would say.
Thank you.
Thank you.
Our final presentation of the forum is going to be a virtual presentation by a team. The lead presenter is Daniela Delicato. She is a qualified person and a life science senior consultant for ProPharma. She will be supported in her presentation by colleagues from the University of Vienna, Dr. Laura Maghini, a senior researcher, and Professor David Bonifaci, a full professor. The title of their presentation is Risk Management Frameworks Vital for Addressing Supply Chain Risks in Biomanufacturing. Daniela and team, the floor is yours.
Just 1 second. Daniela is right here. She's just having some technical issues sharing the presentation. So we'll be back in a second.
Okay, so can you see my presentation? Sorry. Hello. Good morning.
Yes, we can see your presentation.
Okay, okay. Finally, thank you. I win. Okay, and in this mode is better now? Okay, so thank you for the opportunity to be here today. So I want to share useful insight into an important topic, so the complexity of supply chains in biomanufacturing. In recent years, the biomanufacturing industry has grown rapidly. Consequently, the need for innovative, reliable, robust, and compliant supply chain solution has become more critical than ever. Maintaining product integrity, managing cold chain logistics, and ensuring regulatory compliance are particularly vital challenges in this landscape. In this presentation, I will discuss key trends and risks in biomanufacturing supply chains, the role of digital transformation in managing this risk, and the evolving regulatory guidance in cold chain management. Finally, I will highlight innovative solutions that ensure product quality across the supply chain. Let's begin by examining how global biomanufacturing supply chains have evolved. This supply chain is really complex and relies on a complex interconnected network where APIs and drug substances now today are produced in one region and transported globally. Managing this trans-regional supply chain.
May I interrupt?
Sorry.
If you're moving your slides, you were still on the title slide, and it's not in presentation mode. If you could please put it in presentation mode, we could see it better.
Okay, so help me how I can do, because I'm sharing my screen and sharing the presentation, but I don't know how to present. Can you
Yeah.
can you lead the presentation, Amila?
Yes, that will be best. You just say next,
Yes.
please, and we will put next. Thank you.
Maybe we can control.
Yes.
Yes, we will control. Go ahead, Danila. We're ready.
Okay. So the slide number 5, please. Okay, so I'm speaking about the complexity of the global supply chain in today, where APIs and drug substances are produced in one region and transported globally. Managing this trans-regional supply chain presents significant challenges due to the different climate, regulations, and logistical requirements across regions. To manage this challenging supply chain, partners like 3PL, 4PL providers play a critical role in ensuring product integrity by monitoring shipment and ensuring compliance with the rigorous standard at every step. Strict adherence to regulatory standards is essential to mitigate risks such as product degradation or mishandling. This not only protects the integrity of the product, but safeguards patients from potential adverse effects. Scroll on, please. Now let's look at some important trends. Next slide, please. Recent data indicated that the global biotech spending is projected to surpass $8,090 billion by 2028. And note that with the term biotech, we refer to a wide range of products derived from living organisms, their cellular and molecular components, including peptides, antibodies, cell-derived products, such cultured and engineered tissues. Next slide, please. One notable trend is that 70% of new medicines introduced over the next 5 years will be specialty or specialized therapies, which require highly specialized expertise for production and careful handling. For example, in next slide, we can see CAR T cell therapy, which utilize a patient's own cells in a process involving cell collection, processing, and reinfusion, all conducted under strict temperature and time constraint. Next slide. As demand for biologics and advanced therapies continue to grow, the biomanufacturing industry is under increasing pressure to adapt and innovate. This involves addressing intricate challenges in logistics, regulatory compliance, and sustainability. Companies must now invest in innovation, digitalization, and real-time monitoring to meet these growing demands. For therapies requiring highly specialized logistic solutions, such as ultra-low temperature storage, automated system, ensuring safety efficacy is now essential. Additionally, regulatory oversight is increasing, with evolving guidelines demanding more robust compliance efforts. Sustainability is also become a key focus, especially in cold chain logistics, where minimized CO2 emissions must be balanced with maintaining precise temperature control to safeguard product quality. With this context in mind, we can scroll on with the next slide, and let's to see the specific risk associated with this particular supply chain. Next slide, please. The biomanufacturing supply chain is sensitive and complex, requiring precise coordination among multiple stakeholders, and specialized expertise is essential to avoid disruption across every stage, from production to final delivery. Failures in communication or improper handling can have severe consequences, including delayed deliveries, compromised product safety, and adverse patient outcomes. Biomanufacturing product can be damaged by small changes in temperature, we know. To prevent this, we need a reliable packaging, refrigeration system, validated, and real-time monitoring. The entire supply chain must follow strict rules to ensure the quality and safety of the products. Any failure in compliance can expose the companies to risks such as fraud, disruption in product continuity, and potential regulatory penalties. Biomanufacturing products require strict chain management to preserve their integrity during storage and transit. This involves using specialized systems like refrigerated trucks or cryogenic containers to prevent temperature fluctuation that could compromise efficacy. I think that is the next slide, please. The final delivery stage, particularly last mile logistics, is critical, driving demand for specialized and regulated service providers to ensure quality and compliance. Cold chain failure can have a big financial impact. Every year, it was estimated that the biopharma industry faced losses of approximately $35 billion due to cold chain and temperature control issue. This risk, fortunately, can be reduced by using better logistic and digital solutions. Digital transformation, we can see this, the role of digital transformation in supply chain is playing a crucial role in enhancing supply chain control and visibility. By adopting digital technologies, companies can gain full visibility into the product's journey and take immediate corrective action when deviation occurs. In the next slide, we can see an example of digital tools. IoT device allow real-time monitoring of temperature, humidity, and the location. If there is a problem, teams can act quickly to take a corrective action before that quality is compromised. This also helps facilitate a product release by anticipating issues and investigations. Additionally, IoT enables route optimization, adjusting delivery paths based on a variable like traffic or weather, which is especially important when transporting biologic to remote locations. So faster responses to demand fluctuations and supply disruption, making the supply chain more flexible, and this is very important. Blockchain technology creates a secure record of every step in the supply chain, making it easy to track products and prevent counterfeiting. This is particularly vital for high-value therapies, as it helps prevent counterfeiting and strengthens data integrity, building trust across the supply chain. Automated system finally reduce manual errors in efficiency, helping to improving compliance with the regulatory standards and maintain consistent quality and compliance. Next slide, the focus is that digitalization. The message is digitalization is today more than just an update to technology. It brings significant cost savings, reduces waste, and enhances regulatory compliance. This advancement enables biomanufacturers to better address today's challenges while maintaining the highest standards for patient safety and product integrity. Finally, to summarize the next slide, number 515, product traceability is essential for supply chain integrity in biomanufacturing, as told. Technologies like Bluetooth tags, RFID labels, and data loggers offer real-time visibility into package location, ambient condition, and exposure to factors like light or moisture, ensuring quality and safety from production to delivery. Real-time temperature and equipment monitoring is also critical. Wireless sensors detect temperature fluctuations, sending alerts to teams for quick action to prevent risk to product quality. Fleet optimization further enhances efficiency through IoT-enabled route planning, dynamically adjusting shipments based on traffic or weather. This minimizes delays, reduces risk, and ensures precise delivery even in remote area. Next slide, please. Let's look at the key benefits of digital transformation in supply chain to summarize, because we told about this. The integration of IoT and cloud platform is reshaping supply chain management by generating actionable insights. This approach improves efficiency, reduces waste, and ensures compliance with stringent cold chain standards. Continuous monitoring through IoT prevents temperature deviation, regarding product quality and building patient trust. These technologies also minimize spoilage, reducing financial losses and extending product shelf life. Digital tools simplify regulatory compliance, ensuring adherence to cold chain mandates. Most importantly, reliable cold chain solutions create a new global market opportunity by meeting the rigorous requirements of international shipment. By enabling the safe export of sensitive products to regions with strict standards, companies can expand their reach while maintaining a high level of safety and quality. An overview on the regulatory guidance in the next slide. Let's discuss our regulatory standard guide and validate cold chain processes evolve with increasing complexity of cold chain. The cold chain is guided by several key standards, including ISO, which sets specific requirements for transporting therapeutic gels and emphasize quality control to maintain gel viability. In next slide, we can see ATM, ISTA 7E, and the ISPE Good Practice Guide in the next line again. And they all provide validation frameworks for transportation, packaging, freight, ULT, and container resilience, ensuring product stability during transit and storage. In the next slide, we see that in global authorities, among these FDA, we know that establish stringent guidelines for process validation in manufacturing, but this extends specifically to the transportation. However, the trend now today is that the same rigorous standards required in manufacturing must also be applied to the distribution process. Meeting these standards involve, as we can see in the next slide, rigorous component installation and operational qualification for a refrigeration system, followed by performance qualification to test real-world scenarios under extreme conditions. Moving forward now to innovative solution for preserving cold chain integrity across the entire supply chain. And I will now hand over to Laura, who will discuss an innovative solution currently being tested at the University of Vienna. Thank you, Laura.
Good morning, everyone. Can you hear me?
Yes.
Yeah.
Perfect. Thank you very much. So first of all, I would like to thank Daniela for giving me the opportunity to speak today at this gathering, and also to the organizers for the same. As a scientist, it's very interesting for me to listen to what the people working in the biomanufacturing world have to say about our technology, because indeed what we are developing at the University of Vienna is aimed at improving the cold chain processes for valuable materials and things that need to be transported at controlled temperatures. So I cannot see the slides. Can you just move to next, please? Next. Okay, perfect. So as a scientist, and specifically a material scientist, I'm collaborating with chemical engineers and engineers in order to develop new systems that are able to track the temperature of compounds and materials that are being shipped in the cold chain scenario over long distances in a way that can be IoT related, which means Internet of Things. What does this mean specifically? That we can enable the process to be completely controlled through sensors, which are able to not only interact with the person that's monitoring everything, but with the environment that the product is embedded in along the supply chain journey. This means, of course, that there will be sensors included in the system that we are developing, that there's going to be some kind of processor which is able to transform the information gathered by the sensors and then translate them in a way that's accessible to the people that are receiving them. And of course, there's going to be something else that's going to help the people that are actually handling the products to immediately understand if there's a problem in the way that the product is being shipped and moved along the supply chains, which translates as a display. So if we can move on to the next, I will not stress again the importance of keeping the cold chain intact, because of course there's problems arising, both product-wise and economic problems arising from the loss of materials due to not a correct temperature control. And so I'm going to go straight directly into the technology we're developing. So translating what I've been introducing so far into something more like visual, I want to introduce you to our visual smart label, our idea of how things should be made in order to help the process of cold chain control and improvement along the way. What does visual smart label mean? Basically, what we want to do is we want to transform the standard labels that accompany our products along any journey into some kind of interactive label, which is able, as I was mentioning before, not only to gather the information from the environment in which the product is moving, but at the same time also transform the information into data that can be worked on, and then through machine learning, we can understand where eventual failures of the cold chain have happened, but also immediately on demand and on spot inform handlers of these products, especially in remote regions or in places where we don't have internet connection, or if we cannot use a reader, for example, in a -40 fridge, but there is some problem with the product. And we do so by introducing into these labels all the required electronic components, and especially a display, which would be the round shape that you see here in this design. And this round shape basically is an interactive display, an electrochromic display, so very low power consuming, a kind of an e-reader type of display, which is connected to the sensors. And as soon as something is detected, like an anomaly in the cold chain, something like a symbol or whatever other information the promoter or the owner of the product or the label requires will show onto the label, telling the people that there is a problem with the product along its journey. What are the advantages of smart labels? Smart labels can be put directly onto the package, and they're very sensitive to the specific environment each package is experiencing. So you don't have like a bulk data, like something that you put just in a truck, but it's something really related to that specific product. They're tamper-proof. They are, of course, able to reply instantaneously to the changes in the conditions in which the package is moving. And this can be done both online and directly live with the person that's handling the product. This can be specific for each product or can be used for bulk care shipments. It depends on the usage what people want to do, and they're extremely tailorable. We can put any type of sensors, more than one. We can have also pressure, humidity. We can have the tracking of course, the position of the label and the product. So there's really a lot of opportunity there in order to really tailor the product of the smart label to the needs of the producer, the OEMs, or the company that is handling the shipment. And this also comes with an app. So of course you can have all the information accessible also through the app. And of course a cloud system if you want that, where we can also do the machine learning and understand where eventual failures have happened. So with the smart label, we're able to follow the products along through all the supply chain directly, and it can be made also usable by the end user in case these products or drugs would be delivered directly at home. Next slide, please. So how do these smart labels work? As I was telling you before, we can have different sensors, we can have the printed batteries, we can have the RFID NFC chip in order to add this communication and in case also energy harvesting solutions. And then once we have all the inlay electronics, what we do is we package them with our displays, which allow you indeed to have this direct communication with the smart label, not necessarily having to use a reader or even a phone in case you want to use the NFC to communicate with the label through the app. This is important, of course, if you don't have access to the internet, as I was saying, or if someone who has the need to understand what's going on with the product cannot physically use the phone or a reader because of the environment that they're in. So what happens? We can have two types of activation. We can have the activation that's coming through the sensor. So of course, in that case, what we can do is we can have the sensor directly communicating with the smart label, and the smart label displaying the information as soon as the threshold that we have set in our system is overcome. So what happens is that the sensor gives the signal to the display, and the display instantly shows the information on the label. Or we can have also a communication that is actually coming from someone that's operating it. So we can activate it and say, okay, what happened so far? And so we can have like a checkpoint. And then when we give the trigger to the label, it will display any information related to the shipment in case something went wrong. So we can have both the ways of operating it. And this next slide, please. This works thanks to our irreversible interactive colors, which we developed at the University of Vienna, and we patented the technology, which are colors we can change and transition from a state of transparency to a state of colored, and they cannot be reversed because of an engineered chemical transition that we developed, which does not allow any tampering. So as soon as there is a change and there is the signal which is showing, sorry, can you just go back? Showing on the smart label, then we know that the information is correct. It cannot be tampered. It cannot be changed. So there is no way that something that is actually being kept at the wrong temperature will be used on a patient, for example. If you see below on the bottom right of the slide, an example of something that we've done. So for example, we can have the display, which initially is transparent, showing a QR code. So it doesn't have to be just a cross or a logo. We can have any sort of information. We can have a barcode appearing as well. QR code is highly tailorable. We can have all sorts of information appearing. It can be important also for the quality control and also for authentication control of the product that's been shipped. So there is really a lot of opportunity in this technology. And this QR code is the QR code that's going to link you to our webpage. So if you want to have more information about this technology, please do not hesitate in getting in touch with us at the University of Vienna. Thank you very much for your attention.
Yes.
Thank you, Daniela and Laura. I think we have time for a question or two. Mario?
Thanks for the presentation, super interesting. I was just curious with this last slide of yours, how are you handling the different temperatures? So obviously you're going to have environments of -20, then you go all the way to cryogenic. How are you going to handle that with RFID, or are you going to be using a different technology, MEMS or MEMS chips or anything like that to handle the variations in terms of the samples we're going to be tracking?
At the moment, we are working up to minus 40 degrees. We are testing the system with a company called Europlasma for the transport of plasma samples, and RFID is working until that temperature, and also are our displays. So for the moment, we are not looking at other technologies. So we are going to go stepwise, and in case we need to go to lower temperatures even, we will first test our system and eventually introduce our system with new components. As I said, it's highly tailorable and modular. We can really exchange a lot of electronics components. It doesn't affect the overall functioning of the system. So for us, especially as scientists, we don't have any limit. We are happy to try to integrate as many different systems as possible in case we need those. Unless that we will stay with our system, because of course now we know it's working and we want to keep it as simple as possible. So when we need, we will achieve the new system. But at the moment, until minus 40, we didn't need that. And so we are staying with this, which now we know it's operative.
If I may just ask another question, out of curiosity, when the technology is validated with your partners, will that stay with the university, or that becomes a separate entity, or how we go about that?
So we are about to spin off. But of course, whatever we develop, the IP belongs to the University of Vienna. So once we are spinning off as a separate company, we will go through the negotiation phase, and hopefully we will manage to transfer the IP and further develop it on our own. But at the moment, everything is with the University of Vienna, which is also comfortable for us because we can really focus on developing the product without having to think about the costs of the patenting or other things. So we have in plan of separating, but at this moment, we are still intertwined with the University of Vienna, which is supporting our cause and wants us to anyway develop into a private entity.
Thank you.
Thanks.
Any further questions? Thank you, Daniela, Laura, and David.
Thank you.
Thank you very much.
Thank
Bye.
you. Thank you, everyone, for the attention.
Before we break for lunch, I'd like to encourage everyone to attend the final panel discussion this afternoon at 2:20. The theme of that discussion will be future directions in biomanufacturing trends and predictions. So it is a chance for us to wrap up what we have learned over the last 2 days and perhaps agree on what we would like to see next when we reconvene, hopefully in a year's time. So with that in mind, enjoy lunch, everyone.
jednička, jednička mikrofon, raz, dva, tři, čtyři, tohle je jednička mikrofon, jednička mikrofon, dvojka mikrofon, dvojka mikrofon, 1, 2, 3, 4, 5, 6, 7, 8, teďka mluvím do dvojky mikrofonu, vidím tady World Biomanufacturing Forum, 40 minut na monitoru a teďka, když koukám na ten projektor, tak malinko bliká, vlastně jsem si všiml taková frekvence, ten mikrofon je v pohodě? Trojka mikrofon, teďka mluvím do trojky mikrofonu, 1, 2, 1, 2. A teďka mi ten mikrofon spadl, takže jsem ho rozbil. A tohle je čtyřka mikrofon, ten je teda mnohem víc vygainovaný. Když do něj takhle mluvím úplně stejně jako do těch předchozích, tak je o dost víc na hlas. 1, 2, 3, 4, 5, jo. 1, 2, 1, 2, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. Dobrý? Nic mluví. Je tady možná nastavený.
So I'd like to ask our panelists to please join us on stage. I'd also like to confirm that the online audience can hear us well. Art and Cornelia. So this panel discussion is really a culmination and the tying together of all of the ideas that have been presented and that have been shared and discussed over the past 2 days. We've talked about a lot of technological innovations in process excellence. And what I would like to do now is ask our panelists, first of all, to introduce themselves again, especially for the benefit of the online audience, and then ask 2 prompts and give our panelists a chance to answer either one of these prompts based on their area of expertise. And what I'd like to do differently from yesterday is also ask our audience members in the room, as well as our online attendees, for their comments and opinions as it is appropriate. Okay? So let me start. It's an honor to moderate this session. The discussion, again, just to remind everyone, is on future directions in biomanufacturing, trends and predictions. It is a very broad topic. I'd like to ask Cornelia to introduce herself, please.
Yeah, hello, my name is Cornelia Haas. I am from the Manufacturing Science and Technology Department at VTU Engineering. So my core thing is process transfers and process validation of biopharmaceutical processes. And as this is connected to, so closely connected to the biopharmaceutical facilities, now the new topic that I am delving into are energy flows.
Hi everyone again. I'm Mario. I'm the managing partner of Cogen Venture Partners, which is an advisory and investment group based in Copenhagen. We work with venture capitalists, with corporates, we work with startups and accelerators around the bioeconomy, biomanufacturing being a component of it as well. A lot of our time it probably gets spent more on industrial biotech than pharma per se. But there's a lot of commonalities in terms of what we've been discussing here on the biomanufacturing side, and I'm looking forward to the panel discussion.
Thanks. I'm Art Thijsman. I'm the CEO of Biocenter Pharma. We've been developing continuous manufacturing for about 10 years now, and we've been doing that in combination with product development so that we really validate the system in the right way. And now we've reached the point that we think the technology is mature enough, and we're licensing the technology to the industry. So that's basically what I've been doing the last few years.
Thank you. And just Art joined us at the last minute, so I'm very grateful for him stepping in place of the 2 original panelists. So thank you, Art. So let's get right into the questions. The first question is, how do you see process innovation and production excellence evolving in the near term, meaning in the next 3 to 5 years, as it applies to your area of expertise? What specific drivers are the most influential in making these innovations a routine part of manufacturing operations? The alternative question is, there has been an intense focus on digital smart systems and the role of automation and digitalization in the manufacturing facility of the future. Where is the greatest potential for digital smart systems to be deployed in support of biomanufacturing? And if possible, can you cite specific examples of these emerging technologies and how they may evolve, again, in the next 3 to 5 years? So let's start with Cornelia.
Yes, I think I can address both questions.
Wonderful.
Because I'm kind of working on both. So with respect to the process innovations, I think for me, the thing that I was following up now, or kind of what I'm interested in, now also relating to the topic that we presented in terms of the carbon footprint, is now finally this decision between, or well, I don't think there will be a decision between stainless steel facilities versus single use and the emissions from it. And I think it's really interesting how the different stakeholders kind of put up their carbon footprints and then life cycle assessments. And there is kind of at the moment no decision for one or the other. And I hope that this will evolve at some point to a level that that it can be kind of directed towards one or the other. And with respect to the automation, there I think is gonna be a tremendous change, tremendous changes being happening now in the facilities in terms of the data that is gathered. So I think this awareness of the requirement of data gathering is now getting more into the facilities. Like we doing this energy flow analysis, for us it's still a hard time to gather the data and get the awareness. But this is the basic work then to use it not just temporarily for setting up this energy flow systems, but then also to do this on an online basis throughout routine manufacturing. So I have recently heard a talk about a pharma facility which gets kind of autark because the electricity grid is not, the grid electricity is not carbon neutral. And therefore they are based then on solar, wind power, as well as a little bit of biomass incineration. And by the different types, they need to compensate for the availability of wind and solar in their own little tiny grid. And I think this is very interesting to have the thoughts put in, and there also to think about the control systems that need to be adapted then, because they currently they don't, it's not possible.
So is this something that you think is part of a future trend in terms of this type of data gathering and analysis?
Yeah, I think at the moment we're in the stage of just raising the awareness for the need of gathering more data, more online data. And then the next stage will be to utilize this more in processes like what we heard from you, the online measuring system, integrating then into the processes, but also on the facility side, the status of the facility, and tell you when the machines need to be, when the maintenance of the machine should be done, and not do this on a regular basis anymore, for example.
I see. Thank you, Cornelia. Mario, your perspective, please.
Yeah, I'll give my perspective more from maybe from the fermentation component of biomanufacturing, but I think it's going to go into a lot of the points that Cornelia just mentioned. And when I think about innovation will evolve over the next 3 to 5 years, back to the first question, I do see that evolving from a lot of the technologies that we have spent the last few years developing, a lot of great sensor technologies, a lot of elements to start combining and gathering data, and some really good monitoring tools. And I think we're going to start moving that further down the path of process control, and in a way that basically we go from not only collecting the data and monitoring, but also to analyzing, to diagnosing the problems and taking preventive or corrective actions to the process in a kind of real-time basis and in an automated basis, which it's a bit of the holy grail of process control anyway. I think the drivers will they're kind of somehow interconnected, and I think we're discussing that during the lunch as well. The drivers for that sort of innovation, which I think in many ways will be digitally related or digitally driven. And they come from what we discussed yesterday during the panels and doing some of the presentations, which was operational efficiency, which is the core of manufacturing and also the core of biomanufacturing. That's a structural force that will continue to play out. And then the other factor which is related to it, which is a unique element of biomanufacturing, is actually the green transition or sustainability push, where I guess I would be preaching to the choir if I was to say that biomanufacturing is at the epicenter of how the green transition will take place, and will be part of so many different industry transformations and industry evolutions as far as sustainability is concerned, from pharma to energy to foods to new materials, new chemicals, and so forth. And that drive also pushes biomanufacturing itself to become more sustainable. And that comes in many different ways. One is obviously reducing or optimizing your energy consumption. There might be coming from minimizing residual byproducts. It might be sugars, it might be water, or it might be becoming better at predicting end of cycle, so you can conserve more of your resources. So all of those things which have this sustainability angle and can potentially improve your carbon footprint, also automatically go into operational efficiency as well, which is a kind of a nice touch of the industry we're in. And just touching on the second question there, maybe because this flows into it, I think one of the areas where I really see a lot of potential in this process control element is with the sort of smart digital systems or digital smart systems, depending on how you want to call them, which are combining those advanced technologies that we discussed today, but they then combining this with digital sensors or virtual sensors. They're combining this with basically mathematical algorithms. They're combining this with first principle models, Industry 4.0 techniques, ML techniques, all in a way that allows us to start taking action. And there's one interesting technology as an example that I've seen starting to do that, and that has some validation, because I think that's one of the elements that we saw the last 2 days here was there's some incredible technologies. Many of them are still at that sort of development stage and testing stage. And really what we want to see next time and maybe next year is to see some of them translating into things that the industry is adopting.
Yes.
And we have the sort of industry recognition or endorsement, so they can be having a possibility of being adopted. But this particular technology, which is called BioCow, and it's primarily designed, it's a digital suit designed for fermentation monitoring and control. And what they do, which is interesting, is basically they combine these adaptive mathematical algorithms with preventive parameters, with phenomenological mass and energy balance models, to really turn real-time fermentation process data into actionable control measures. And going back to process control and how we take action, it's a really kind of smart way of somehow empowering process controllers with by enabling this kind of seamless real-time process corrections. And I think that's one of the examples. Obviously, that's specific to the fermentation side of biomanufacturing. But I think we're going to be seeing more technologies like that evolving, and that's really leading to this true process optimization that we're looking for, which eventually might take us to production excellence, if we can ever claim that.
Right. Right.
And the funny thing is, or what I'm wondering about is how can these systems be validated?
I know it's and these are not systems that started overnight either. So I think we're also finding out that even if you're using digital tools, this is not just a clear launching an app on the App Store and having exponential growth overnight. This particular technology, they've been working on that for 7 to 8 years.
Yes.
So way before and they don't even like to talk about ML, even though they use ML techniques into it, because they kind of, we don't want to be caught up into a particular hype, because that's not how we're selling the product. And they spend many years working with. They're basically deployed in 7 different fermentation sites, and they have another 5 or 6 that basically doing a POC with them. They'll be across biofuels, biopharma, and brewing, or different fermentation processes. And but it took them a long time, and they had pushback, and they tried with different customers, and they had to go back and improve the system. And I think that's there's a lot of getting the feedback until you find your proper market fit, and finding you actually being able to validate that. It was only after one of their customers who was actually, look, this is working for us. We can see the results, but we still don't believe it. So we're going to hire an independent party to validate all the results. And they go through another season just doing that. And then finally they got that sort of independent validation. But it's a long journey to get into it, so it doesn't happen overnight. And only then they start opening all the doors, because then their competitors start looking and say, well, that's something that, then it creates a bit of that formal element of, well, our competitors are using, maybe we should be using this as well, because they're getting yield improvements, they're getting energy savings, and so forth. But it just takes time, and I think you have to, as much as we like the element of digital being a faster paced element for us to develop it, you still have to go through the motions of being in a slow moving industry where things take some time for innovation to be adopted.
That's right. And I think the key here is to show small wins at the beginning. There's this grand vision, and we get lost in the vision without realizing that there's all of these pragmatic aspects like validation of a system that is perhaps iterative, right? Again, not something that would be solved overnight, but something to keep in mind. Now, continuous manufacturing, of course, relies on automation a lot. So, Art, we'd be very interested to hear your perspective.
Yeah, maybe to follow on with the previous comments. Yeah, in general, I'm always terribly disappointed with the speed of introduction of technology and the uptake of new systems. It always takes twice as long as you expect, and there are only, let's say, these companies willing to take risk that actually create the breakthrough. And I'm experienced with our technology, continuous manufacturing. Getting that out into the field is a major challenge because people have to see the benefit, and they want to see proof done by someone else first. And there's always a reason. There's a regulatory problem. Yes. So don't you come back next year. And so in general, I'm always disappointed about the industry, but the solution might be the digitalization. And because that's coming on so strong, the AI component that everyone seems to embrace, I think it's a little bit hyped to a certain level, but that doesn't matter if you can use that to accelerate innovation. And I think that's going to be the case in a lot of areas, that thanks to, because what you're talking about, new fermentation systems, you're talking about a more advanced data acquisition that needs to be done. And yeah, this is something we experience on a daily basis, because if you're running a process continuously, the amount of data that you generate is enormous. And yeah, what do you do with that data? And how do you interpret it? How do you control your process, et cetera, et cetera? So, and there's a lot that you don't know, and then AI can help a lot, machine learning can help a lot. So I'm more optimistic for the coming 3-5 years than I was for the past 3-5 years with respect to implementation. And I think one of the drivers that you're asking for, especially in our area, is cost of goods. And the cost of goods is becoming, we started off also with the biosimilar development, and there it's of course important, but it's sort of flowing over to other areas as well. The cost of goods is a major, and then sustainability comes into play, and everyone thinks that'll cost more maybe, but actually it should cost less if you do things right from the beginning. So there'll be a sort of a mix, I think, the coming 3-5 years of more digitalization, much more data processing. Sustainability will come in a lot stronger from different angles. Especially the big pharma will drive sustainability, I think, more than others. and CDMOs will be more, let's say, adapting to what the client wants. But the biosimilar group will be pushing heavily for lower cost of goods. So I think it's a relatively good time for innovation, especially when you have a high level of a digital component to it, like the analytical part, and then you can go a lot faster. Mm-hmm. Things like new column material, like let's talk about I think he's Sian, his name is, from New Zealand.
Right.
That takes a lot longer, in the sense that before the industry adapts, goes away from classical, Cytiva beads with protein A on them, that takes 10 years or something like that before they really change. But new developments, new products, the adenovirus products, there are opportunities with new developments to bring in new technology. And with the digitalization, that should go faster. Maybe the regulatory people can't keep up with it. So there's maybe a comment you can make, Max, how to tackle that, to get the regulatory people to move faster. But I think there's a great opportunity to push things through the coming years. So push hard on innovation, and that we can make bigger steps than the last 5 years.
And I think one of the major things to drive it, I think, is to see this sustainability or the digital transformation, to see this as an opportunity. Yeah. Because this is a positive motivation, and not something that you have to do.
That's right.
Not the push, but the pull.
Yeah,
that's right.
Yeah, but you notice the industry is very much, there has to be a clear reason to change, and that reason has to be heavily motivated, and the whole plan to implement has to be there. The regulatory people have to agree with it. The clients have to be there. So it's a very regulated industry, which makes introductions per definition difficult. But you have to see the opportunity and take the risk. I agree. Yeah. Absolutely.
For example, in Austria, Russia just kept the gas supply to Austria because of contract issues with our local gas supplying company. So as it seems, as of January, Austria will not be supplied with Russian gas anymore, and this can be seen as a threat or as an opportunity.
Yeah. No, but you have to have that positive, and I think people in innovation always have that sort of positive mentality, and that this is better than the existing, and it should be done as soon as possible. That's the general mentality in the field of innovation. Yeah.
And a lot of innovation is driven by the environment in which we operate.
Yeah, absolutely.
So let's not forget that.
Environment. If you listen to this conference for let's say the climate change conference that was held, I think last week, the weeks before, then you sort of question mark the move towards a changing environment. So it's a difficult field to play in, the globalization field, but I think you should make it smaller in general. What's your area? What are the challenges? What are the opportunities in your area? And just forget the rest and go for it.
Right.
If you know that it's good, just go for it.
But on this topic then of sustainability, how do legacy companies with these aging plants reconcile the need to go green, if you will, with the fact that they have all of these investments in plants that are 10, 20, 30 years old?
Honestly, my first approach was to try to tackle kind of the consumption side and try to reduce here and there and there and there, and then to get less, because the major energy input is still most of the time natural gas. And I just came from the ISPE sustainability seminar of Belgium, and there several large multinational national companies kind of told there, we're not talking about the getting here a little bit better, there a little bit better, but they were talking about really replacing the natural gas. And I think this was, to me, an wow effect.
Replacing with what?
Replacing with electrical boilers, so that you base it on electricity, or with biomass. But biomass doesn't really work for large facilities, only for small facilities, because otherwise you use too much wood.
But electrification, of course, relies on other sources
of
energy. So,
and it's that's why this development needs to go in hand with greener, with a sustainable electricity production. And this can be done. So this can be done by manufacturing or producing electricity on site, which I have seen now at the ISPE. For example, Takeda in Lessines is now putting up windmills besides the solar panels on the roof, which many producers are installing. Or what was it? Pfizer in Puers is buying offshore or is building offshore electricity windmills. So there, and I think this is interesting that the pharmaceutical manufacturers move a little bit outside of their core business of just producing the medicine to taking also the kind of surrounding responsibility of, okay, where does the electricity come from? Because the example what we brought, this is Austria and Switzerland. There it's easy to buy electricity which is free of CO2. But here in the Netherlands or also in Belgium or many other places of the world, at the moment it's not.
Absolutely.
And that's why the pharmaceutical producers take on or need to take on responsibility also for the electricity to some extent at least.
And I'm not thinking just Europe, but outside of Europe, Asia Pac, for example. Completely different landscape.
Well, it's the capacity of renewable energy installed in Asia is much larger than in the rest of the world. So the absolute capacity it is more, it's just a fraction.
Maybe the climate change will, let's say the temperature increase will help there. If it goes up more the coming years, that people will, okay, now I have no choice, I have to go more sustainable.
I think there is just on the sustainability innovation, kind of how we use that. I think when we look at larger corporates, and might be pharma, might be industrial biotechs, they're basically biomanufacturing. One key component for them existence is their own corporate sustainability reports, right? And innovation is actually a very easy way for them to show they're doing something.
Well, that's a good point.
Even if they're not doing a lot. Obviously, less friction, the better. So if the innovation comes, going back to your point about all the plants that needs to be upgraded somehow, or just to make them more efficient, as long as those types of innovation, and it's very different if you were starting a new site where you can basically use new technologies, or you can use new bioreactors, and you can use all those amazing new technologies. But when they look at their own existing footprint, which is a key component of the CSRs, as long as you don't have a major capital investment requirement, as long as you don't have to make some significant retrofits to your existing infrastructure and assets, and ideally, as long as you don't have to change your manufacturing protocols or compliance protocols, I think there is an angle that should be used. I think it's more a function of them finding technologies that they want to be deploying. Going back to the original point about validation, right? And so they're not going to try innovation in their plants just for the sake of it. But as long as something has a means for them to tick those two boxes, which is sustainability and also gain some brownie points on productivity, that card can be played. And I think that's an opportunity that we have because they're kind of running out of options on how to do that as well. So many of them invest in companies just for the sake of saying that they're doing something on CSR. And I think as we evolve with those technologies and might be digital, I think there's an opportunity for that to be used as anchoring for those innovators to say, hey, this is actually going to, every time I work with a company, this is actually something very high on the agenda when you're presenting it to a larger corporate, because that's sometimes even more important to them than the economics, which, you don't want the investors to know that, but that's how important it has become for some of them, because they are held to a certain standard, and a lot of the KPIs of executives are based on those CSR KPIs as well.
That's right.
Yeah, nevertheless, I think we're in a sort of in-between phase, in the sense that, you have the opportunity, if an innovation, let's say, feeds more sustainable, feeds more, let's say, cost reduction, has a multiple advantage. That's the phase we're in now, I think. Then it's adopted. The real bigger steps still have to be taken by a lot of companies, and that still will happen in the coming probably 10 years, something like that. But it's always in movement. But my experience is that if you can get a combination of multiple factors, like, for example, in our case, we have continuous manufacturing. You use your single-use parts 3 times, 4 times as long as in a batch process. So you can claim it's more sustainable.
That's right.
But it wasn't the objective initially to do it that way. But if that makes it easier to adopt, if that's more important on the agenda of a big pharma, and that moves things forward, great. Let's make the combination. Why not? So there, I think, is the opportunity at this moment is making combinations, and nevertheless, innovation is always, per definition, will move, and the digitalization, as said before, that's a real game changer the coming years, and that'll push a lot of things that we probably didn't realize that they were possible.
I agree.
For a few years back, and because if you believe the big AI gurus, they say it's dangerous. AI, I haven't seen anything dangerous yet, but it'll have to have an enormous impact if they're saying that.
Right.
So AI developments will go a lot faster than we've been used to. Automation developments. So that'll be interesting to see that impact. Yep.
Thank you, Art. Are there any comments or reactions from the room or online? No? Okay. One thing that I'd like to address, Art, is you talked about the regulatory aspects. We saw yesterday that the FDA especially has the Office of Emerging Technologies and several pilot programs for critical therapeutic areas, where I think they're looking for specific examples to pioneer the adoption of these innovations, whether it comes from the process side or the clinical side or the operations side. So that's one way, I think, where regulators are committed to advancing new technologies faster than they have in the past, number one. Number two, since we've been talking about modernization of facilities and equipment, I think that there is a lower regulatory barrier to adopting these sustainable and green technologies, because from a facilities and equipment perspective, there isn't as much friction when it comes to regulatory concern as there would be for making significant changes either to the manufacturing or the quality and testing aspects that the regulators are used to seeing.
Yeah, I agree. That's I think and especially the FDA is more active in stimulating technologies, even though PAT I think they almost started it themselves a number of years back, but that's like 10 years ago, and now it's at this moment it's more coming into play.
Yes.
So it takes a long time, even though they're positive towards it. So there are the regulatory bodies in general don't move fast. The FDA, I think, are more open for new developments, but there's still they form a major hurdle to speed up things and to reduce development times. And for example, in the biosimilar field where we are operating, the phase 3 that's done by all biosimilar developers, it's stupid. I think 99.9% has been successful.
Yes.
Phase 3.
Yes.
So and the data packages are so extensive.
Yes.
So just accept that you can do it with a phase 1, and take that jump. And that's something they don't dare to do because there are always these exception products that are more complex and they could form more risk. And so it's going to be interesting how, if they take that into account, because it's being debated for about 5 years now.
Yes.
So kick it off and let's move.
That's right.
With lower cost products. And it'll also be interesting in the ATMP area with the cell therapy, because that's going to explode the coming years. And to see how they react to that, because that's such a logistically intensive setup that's being implemented, and automation will have to come in very heavily there as well, and single-use development. So there's a lot for the regulatory bodies also to, let's say, to change. But that's not their style, changing. So it's a pity.
But it's cell and gene therapy products, the phase 3 studies are way smaller than for conventional medicine. So maybe this is also a window of opportunity to translate back to conventional medicines.
Yeah, but the cell therapy is of course something different than biosimilars. But
right.
The cell therapy, I'm actually surprised that they haven't made much more development than they have till today in the way they planning to do it, because patient cells will be transported to their facilities, will be reprocessed, manipulated, and transported back and to the patients. And the therapy is very effective, otherwise they wouldn't do it. But the costs and the logistics involved is enormous. And you would expect that, anyway, I would hope that authorities it's just like my disappointment on the slow business is also a little bit disappointment in the regulatory not being proactive in general.
Do you think that COVID made any differences to this?
Yeah, of course. For the vaccine world, for sure. But that's what you see. There's a crisis, and then all of a sudden things start changing. New technologies implemented, mRNA within months to half a year, and regulatory bodies, they approve things within. Normally it takes years. They do it in a few months.
Right.
Of course, it's required, but all of a sudden they can do it.
Yes.
So why not change the business model?
That's right. That's right.
And make it more efficient.
Instead, they revert to the status quo. But you're preaching to the choir here. Yeah. And just to add to your point regarding biosimilars, in theory, phase 3 is not required. The WHO guidelines very clearly state analytical comparability, maybe some animal studies, and a phase 1 PK/PD.
Yeah.
That's all.
But it's also the producers said they don't dare to take that risk regulatory-wise. There are not that many that do an application with only the phase 1 and the data package. So it's also the other side as well. But
Perhaps it's choosing the right regulators. Roman and I had this conversation earlier. Maybe there are regulators in emerging markets, and LMIC is willing to take that risk. Who knows?
Yeah.
Just throwing that out there.
No, but in the UK, that's where they've officially announced that the phase 3 is not required.
Yes.
For the regulatory bodies.
Yes.
But I haven't heard of any product yet going through the whole system.
Stay tuned. That's all I'm going to say. Any other comments? No? Okay, maybe I can ask the last prompt. Well, we already talked about this. I was going to ask what barriers do you foresee in the implementation of these future innovations. We talked about that already. We've talked about how manufacturers, regulators can work with stakeholders to make these innovations a reality. I don't think I have anything else that I would like to expand on. Any final thoughts from the audience? No?
Maybe one last comment, because there are a number of very early technologies that were presented today.
Yes.
And I really I'd like to boost these people to continue
Yes.
with that work, because it's really important, and it really makes my heart important. And we're talking about manufacturing and all that kind of stuff, but we have to keep feeding it with R&D.
Yes. Yes.
Yeah.
I fully agree, and as I mentioned earlier, perhaps there's an opportunity next year. I'm looking at you, Mila, to get updates from several of the presenters that we have hosted. Can report something tangible that has happened over the previous 12 months. So that constitutes, I think, my closing remarks as well. I have nothing further to add. Again, on behalf of the organizing committee, I would like to thank all of the attendees. A special thanks also to the panelists and the speakers for taking time out of everyone's busy schedule to convene face-to-face. I'd also like to acknowledge the folks that are online. Thank you for joining us, and we hope to see everyone next year with both old topics and new. Thank you.
Thank you.